Testing, testing. Students, can you hear me? Online people, can you hear me? Yeah, can you hear me now? 5 more minutes maybe for the paper. Okay. Mm. While you're doing that, did I give any homework? Any homework? All right. There was an essay to write, right? Okay. We'll, I mean, together let's make a marking scheme for that. Right. Then you will have an understanding of how to write essays and also how to mark your own essays. Right. That's a skill you have to develop now. So, online students, are you done with the paper? Yeah, it's raining here, so there might be signal issues. So let me know if there's any lag or uh, any connection issues. Right? Okay. Who's new today? You're new today. So this is last week's paper. You can just go through it. Yeah, paper three. Okay. So you are new, right? So this is last week's paper, so you can just go through it, or you can watch the recording and just get the answers. It's not that difficult, right? So it's just given to you to just get up to speed of what you have done, what we did last week, right? So you have to study, right? Not by studying, I don't mean like you have to by heart it completely within that week, but you have to be in touch, right? You can't forget what you learned previous week. Otherwise, it's going to be it's going to defeat the purpose of coming to the class, right? Especially for bio, you have to keep in touch. Like have a small idea what you learned last week. Have a small idea, right? Otherwise, you will forget what you even understood, right? The concepts. If you forget the concepts you understood, that's a waste. You don't need to memorize everything. Right? You don't need to know everything by heart within 1 week. But you have to keep in touch with the concept. Otherwise, it's a waste, right? Then it's a waste of the theory class. Because why? Whatever you need to memorize is there in the resource. So that's going to defeat if you have forgotten what you learned, right? So keep in touch with what you learned. What are the different types? So, for example, last week, what are the different types of properties? What are the properties of water that gives rise to those special properties? Then, what are the classifications of carbohydrates? How can we classify? Like that, have the sort of the outline of the concept in your head, but not everything, right? Not exact function. If they ask you inulin, what is the exact function? You don't need to remember exact location, but just remember it's a polysaccharide. Then right, have a general idea every week. That's my sort of idea of giving this paper. Right, online students, any issues because of the rain? Do you feel any difference in the video? No, right? Do you feel any difference? No, right? Okay, right. Okay, done with the paper. Yeah, done. Okay, online students, I hope you are done. Send me some answers in the chat. Right. Okay. First question, which of the following is the incorrect combination regarding carbohydrates? So underline incorrect. So in the level paper, it will be sort of bold, right? It will be bold in the paper, right? So in here, I didn't go to the trouble of bolding it, but always pay attention to what they're asking, right? If they're asking incorrect or no, right? Those are silly mistakes. ශිෂ්‍යත්ව කාලෙත් මේ කියන්නේ මේක incorrect ද correct ද කියලා බලන්න කියලා හොඳට, right? So in the LOL paper, it will be highlighted. You will net. It's very unlikely that you will miss it, right? Incorrect කියන එක දහන්නේ කියන එක. Okay. So transporting plants sucrose. Right. Sucrose is what? Is it a mono, di, or poly? Sucrose? Yes. Sucrose හදන්නේ මොනවාගෙන්ද? What are the two monosaccharides? Glucose. ඔක්කොටම glucose ඕනේ. For any disaccharide you need glucose. What else? glucose and fructose, right? So transport in plants. Did I talk about this in class as a function? No, because why? At the end of the class, we didn't go through the carbohydrate function table, right? So that's what we are going to do today. So under the functions of disaccharides, if you come to the resource book, turn to the resource or the tube, take out your tubes, online students. You got a tube? Oh, okay. It's fine. Just okay. So in the end of carbohydrates, we have a table that we didn't go through, right? Online students, take out, turn to the PDF of the tube, right? Or if you don't have it, then you can come to the resource book, right? So there we have the separate functions of monosaccharides, disaccharides, and polysaccharides tabulated, right? Jump to disaccharides, and under disaccharides we have what? Sucrose. Yeah. Okay. So functions of carbohydrates, can you see under disaccharides we have uh, three functions of disaccharides. Can you see page 12 of the resource book if you are using or in the tube, right? Yeah, got it right. So what are the two disaccharides that you need to know with their functions? First one is lactose, right? Lactose, even though it's a disaccharide, right? So what are the properties of a disaccharide? Yeah, they are still considered as sugars, right? Disaccharides are still considered as sugars. Yeah, why? What is the basic property of a sugar? Sugars must be sweet, right? So disaccharides are very similar to monosaccharides in properties, in terms of properties. They are sweet, right? They are crystalline. They are crystalline. Yeah, third one. Any suggestions? Water soluble? Yeah, that is again a similarity to monosaccharides. Water soluble. And also, there's an additional thing that monosaccharides don't have, that the monosaccharides can't be broken down further, right? Monosaccharides is the simplest form. No? So monosaccharides can't be broken down, but your disaccharides can be converted, or what we call as, what do we call the process of breaking down a disaccharide to a monosaccharide? Yes. So how did we make the disaccharide? By taking two monosaccharides and removing a water molecule, right? It's called a dehydration reaction or a condensation reaction. So we have to do the opposite. We have to do the opposite. To disaccharide to monosaccharide, we have to add water, right? What is the process of adding water called? Mhm. So if removing water is dehydration or condensation, this is called hydrolysis. Hydrolysis. Okay? So disaccharide, when we add water or if we hydrolyze it, we get a monosaccharide actually. So how many monosaccharides? Monosaccharides times 2, right? So coming back to the functions, in sugar, so in milk, you have a disaccharide that is actually serving as a storage. But disaccharides are not very good storage molecules. Why? They are water soluble. If you store something somewhere, right, and take a plant, right, take a plant, right, if there's water everywhere, right, in a plant cell, inside the plant, there's water everywhere. So if you store something somewhere and that something is water soluble, that's going to be a problem, right? It's going to get dissolved in water and it's going to go away. That's why plants store what as their food? Starch, right? They store starch. What is the most important property of starch? It's a polysaccharide. So it's not water soluble. So ඒක තියපු තැන එයා හැමම ඉන්නවා, right? Take like a අලගෙඩියක් වගේ එකක්, like a tuber or something. It's composed of starch, but that starch is water insoluble. So it will stay there, right? But some disaccharides are considered, I mean, yes, they are storage. So the example is in milk. You have a disaccharide that is lactose, that is a storage sugar. Okay. Then in sugar cane also, sucrose has a storage function, right? That is sucrose, right? Sugar cane, you know. Give me another example of a plant that has sucrose as storage. Have you heard? ලංකාවේ ඒගොල්ලන් අපි ගන්න වැඩිය සීනි විදියට extract කරන්න. Our main sugar source is sucrose, sorry, sugar cane. In other countries, there's beet sugar, beetroot really, right? Because sugar cane is sort of a tropical, right? අපි නිකන් මේ ඉන්දියා ඒ පැත්තේ ලැබෙන plant එකක්. So in Western countries, to extract sugar, they use beetroot, right? Beet has a sucrose, a lot of sucrose storage as something additional. Third disaccharide with a function is again sucrose, that is the transport function, translocation, right? Why do we call it translocation, not transport? That's something you will understand very clearly when you're doing plants, right? In phloem, right? Phloem is a sort of a very variable thing, right? There's no definite direction to phloem transport, right? One day, some part of the plant will be producing phloem and going somewhere, and another day that same part will be receiving phloem. Yeah, it is variable, right? So phloem movement is a sort of a bidirectional. It can go either way. So we use a special term for that called translocation. Okay, so remember sucrose is the sugar involved in translocation. So better correct this, right, in your paper 3, cut the transport, right, cut the transport word and say translocation in plants. Better write phloem translocation, okay? Phloem translocation in plants. Online students, you can hear me, right? Okay. Then second one, component of middle lamella, right? We talked about the middle lamella, right? It's like the cement between plant cells, middle lamella, right? So middle lamella is composed of pectin, yes, right? Middle lamella is the name of the structure. What is the constituent, like the I mean, what is it made of? Pectin, okay? So that is also true. Storage sugar in milk, we just talked about this, that is true, right? Lactose is a storage sugar that we consider. Transport in humans, glucose. Glucose, well, what is the transport method inside our body? What transport method do we have inside our body? Our blood circulatory system, right? So in the blood circulatory system, do we have glucose going everywhere? Yeah, Do we have glucose? We have, right? Have you done like a glucose test? Right? It's called capillary blood sugar. Then you put it on a sort of a glucometer, then you test. So in a given time, you have a lot of glucose, right? So right now, maybe, right? You just had breakfast within 1 hour. So maybe you are having like the spike of glucose. Then the glucose will say, you didn't get the paper, right? So, yes, glucose is circulated. You got it. Glucose is being circulated in our throughout our body at any given time, right? So that's a correct statement. Transport in humans, glucose, right? Structural component in fungi. Hmm. What's given? Glycogen, right? What is glycogen? It's a so they're asking structural component. So if we are thinking going in that route, structural, that means like what? So we are classifying based on function, right? So structural means a function, right? Polysaccharides, what are the two ways that we can classify? We can classify them functionally and like in architecture or like how they look like, right? So functionally and structurally we can classify. So if we functionally classify, what are the two types? Storage and structural, right? So a glycogen is a storage polysaccharide, right? Glycogen is a storage polysaccharide. Okay. So we can't consider it under the structural category. Okay. So that's wrong. Write a review in front of it. Write a small review. Glycogen, glycogen is a storage polysaccharide. Is a storage polysaccharide. Storage polysaccharide in what? Animals and fungi. Animals and fungi. So there, can you see that's how it's going to be confusing because fungi has a sort of like a diverse variety of polysaccharides in them, right? They have glycogen that is similar to animals as well as the storage, right? So fungi, glycogen is a storage in them. So if we come to the structural component in fungi, how can you correct this statement? So glycogen is not a structural component in fungi. What is the structural component in fungi? Yes. What is the component of fungal cell wall? Chitin, right? Then write a second statement under that. Chitin, chitin, chitin chitin functions as a component in chitin functions as a component in fungal cell wall. Chitin functions as a component in fungal cell wall. Okay. Right. So first MCQ done. Right. Which of the following, second one, which of the following is a polysaccharide made of, right? Made of aldoses, right? Hope you remember. How can we classify carbohydrates? So we can classify sugars into two types as aldehyde sugars and ketose sugars, right? Aldehydes and ketose. So what are the examples for aldehydes? Yes. If we come to think about the monosaccharides, what are the examples that contain the aldehyde group, right? Glucose and What are the aldehydes? Glucose and galactose. Yes. Who is the ketone sugar? Who is the ketone sugar? If you consider monosaccharides, fructose, right? Fructose is the ketone sugar. Okay. So using that knowledge, you can answer, right? Aldoses. Oh, so glycogen. So we have to go much deeper into that, right? So we are what are we given? We are given polysaccharides, right? So in order to find out the answer, what do you need to know? You need to know what are the monosaccharides of this. So in front of each one, can you write the monosaccharides? Glycogen, what is a monosaccharide? Israelian. What is a monosaccharide of glycogen? Glucose. Right? What is the monosaccharide of hemicellulose? What is hemicellulose composed of? Variety of pentoses and hexoses. Pentose and hexose sugars. Inulin, what is the monosaccharide? What is the monosaccharide of inulin or monomer of inulin? Yes, fructose. Yeah, fructose. Cellulose, what is the monosaccharide? Glucose again. Pectin, pectin, galacturonic acid. Yes, galacturonic acid. Right. So now you have established what are the monosaccharides. Then what do you have to classify? Just the aldehydes. You have to just decide what are the aldehydes. So what are the aldehydes? You have glucose, right? So what are the things that are composed of glucose? Glycogen and cellulose. A and D. Hemicellulose is actually mainly composed of pentoses, so you can't consider, right? Then inulin, can you consider? It's a what? Fructose is a ketone. So in front of that fructose, right? Ketone sugar. Ketone. Okay. Second MCQ clear? Yeah. No issue. Right. Third one, which of the following polymers is found only in plants? Polymers found only in plants. Inulin, hyaluronic acid, glycogen, maltose, chitin. Right. I didn't talk to you about hyaluronic acid, right? No. Hyaluronic acid is sort of like our pectin, right? What is pectin now? Is a cement between plant cells. Yeah. There's a middle lamella and plant cells are pasted together by a middle lamella, right? That's composed of pectin. What about animal cells? What about animal cells? Aren't our cells connected by a cement? Right? There is. So our cement is called hyaluronic acid. Okay? Hyaluronic acid is actually composed of pentoses. Okay? I'll give you something bit of a fun thing, right? So you know our egg, no, right? The female ovum, it's a massive cell. It's a massive cell. The egg produced by a female, right? It has sort of a massive outer protective layer, and it has like a whole group of supporting cells. මේම වටේට ඉන්න එයාට සේනාවක් වගේ supportive cells. This is an egg, okay? Or we call it as ovum. You get it, right? A female egg. So who is going to try to get into here? මෙයාගේ ඇතුළට ring ගන්න හදන්නේ කවුද? During fertilization? Who? Sperm. A sperm is going to try to sort of drill into here, right? So sperm is kind of like this. It has a head and it has a body like this, and it has a long tail, right? So this is a sperm. So this sperm is going to try to get into here because the female DNA is going to be here and the male DNA is sort of somewhere here. Okay, you have to combine the two, right? But the problem is the egg is protected by a lot of cells. right? එයාගේ නිකන් ආරක්ෂාවට imagine like a princess surrounded by එයාගේ servants, right? So the egg is like that. Because why? The egg has to be protected. The egg is released from the female's ovary and it comes down slowly through the fallopian tubes. You have heard of these, right? Right? එනකොට it has to be protected. Why? Because there's only one egg per month, right? There's only one egg produced per month. No? What about sperms? During one like ejaculation, we call it ejaculation, right? A male releasing sperms. Do you know the count? How many sperms are released in one ejaculation? Per milliliter of sperm, you have 2 to 4 million sperm, I think, right? Per milliliter. And the average ejaculation volume is about, I think, 4 to 10 milliliters with release a male, right? So if you think, let's say, average 2 million into, let's say, 4, about 8 million sperms will be there. So sperms ගැන වැඩි හිතන්න ඕනේ නැහැ. There's ඕනකට වඩා sperms තියෙනවා. But මෙයා protected not, right? That's why it is protected by this sort of other cells. So my point is, these supportive cells are connected by that hyaluronic acid, the cement, right? The cement. Okay, there's hyaluronic acid connecting all these cells. So how can this sperm sort of get to here? Problem, right? It has a weapon. It has sort of like a drill. What can it do is, here on its head, he has an enzyme. He has an enzyme called hyaluronidase. Okay, this enzyme can break down this hyaluronic acid, right? එයාට පුළුවන් මේ hyaluronidase කියන enzyme එක release කරාම it can sort of drill through this cell layer, supportive cells ටිකෙන් කොහොම නිකන් it can separate them and come inside. It's like a chemical weapon. Sperm එකේ head එකේ තියෙන, right? So my point is, hyaluronic acid you will learn about when you are doing reproduction. We have to study a lot in detail about the structure of the egg, ovum, and how the sperm comes inside. Okay, it's in your syllabus. So hyaluronic acid, what's the question? Found only in plants, right? Never. Hyaluronic acid is never found in plants. Glycogen, found only in animals and fungi. Glycogen found only in animals and fungi, right? Maltose, where do you have maltose? It's not in your syllabus, right? But in O levels you learn, right? Where do you have maltose sugar? Maltose is a disaccharide. Where do you have maltose? Yeah, right. Germinating seeds, right? Have you heard of mung seeds? When they are sort of put in water, their starch will be converted to what? So their storage sugar is starch, right? In a seed you have starch. The starch will be converted to maltose, right? So maltose is actually it doesn't have that much of a function, but it's anyway found in plants, right? So which of the following polymers is found only in plants? Then chitin found in fungal cell walls and arthropod exoskeleton, right? Karapottage වටේ කර ගාලා when you step on like a dead one, maybe අර පිටිපස්සේ එළිය තියලා යනවනේ exoskeleton එක like cage එක වගේ කරකරස් ගන්න. So chitin is like crispy, right? So that is chitin. Okay, so found only in plants. What do you think? What's the answer, Puta? What answer did you get? Inulin, right? Inulin is the only sugar, polysaccharide that is found in plants. It's found where? In Dahlia tubers, right? Any questions? So maltose right? Maltose have you like අර පොඩි පාන් කැල්ලක් වගේ අරගෙන කටේ හප ටික වෙලාවක් හිටියොත්, right? There's an enzyme in our saliva. I told you about this, right? Our saliva has an enzyme. So එතකොට bread වල ඉන්නේ කවුද? Starch, right? You can write this reaction. Starch will be converted by salivary amylase inside our mouth to maltose, right? Inside our mouth itself, it will be converted. So there in front of maltose, you can write this. Starch will be converted to maltose by salivary amylase. Write down in the paper. Starch converted to converted to maltose by salivary amylase. Digestion starts in our mouth. Okay, කටේදීම පටන් ගන්නවා. Okay. So where can you find chitin? In front of it, right? In front of chitin, where can you find chitin? Fungal cell wall and arthropod exoskeleton. Fungal cell wall and arthropod exoskeleton. And tell me something additionally special about chitin. Like if you take the molecular structure, something special about chitin. It has nitrogen, right? Which is very rare for a carbohydrate. Okay. What are the basic components of a carbohydrate? Carbon, hydrogen, oxygen, right? Those are the three elements that we consider. But some situations, such as in the case of chitin, we have nitrogen. Okay. You can write down within brackets, chitin has nitrogen. Okay, come to the structure essay. Give me 1 minute. Okay. First question, state the major property of water which helps small insects to live on the surface of a pond. Not to live actually, walk. Those insects don't live on the surface of water, right? අපි ඒගොල්ලන්ට කියනවා water skates. Have you seen අර pond එකේ surface එකේ ඇවිදින insects ලා? Right? They can walk. Right? I told you drew like the how structure of the foot is, right? So water skates. Major property of water is what? Surface tension, right? Surface tension. So cohesive behavior, does it say that? So yeah, this is the closest property is surface tension, right? Major property. So don't define it like that, but you have to be specific in that sense, right? So cohesive behavior you can apply in other situations, such as the ascent of xylem, right? So cohesive behavior is not like a specific thing to walking on water, right? So whenever the walking on water comes, the answer that has to come to your head is what? The surface tension on the surface, right? That's why you can walk. Then you can come to like you can say hydrogen bonding as well, right? So right, so you have to be specific. What is a specific property? Surface tension. Yeah, I see your point, right? It comes under the major property of cohesive behavior, but the specific thing is surface tension. Okay, what is the additional polar group? Oh, you can't do that. Okay. Right. Those two questions you can do. After today, we can do, right? And after we have done phospholipids and also proteins, then we can do. Okay. Those two structured keep. Okay. Online students. First structured essay question, part 2, A and B, you can't do. Come to the second question, define metabolism. Yes. Do you remember the definition of metabolism? What is it? Yeah, tell me. What is in their resource? Sum of even anything? Sum of I can't remember. What about that? I have to check. But you have to remember, me come on, 2 weeks ago, what is metabolism? Sum of all chemical activities taking place in an organism is metabolism. Sum. Okay. So can you see it has two rules given there? That means what? They are expecting something additional. So in a structured essay, right? In a structured essay, you have to sort of read the mind of the person who has made the paper, right? How are they giving you a hint? කොච්චර size answer එකක් දෙකලන්ට ඕනේ කියලා. By the amount of space that you are given in the structured essay paper. Right? Rule දෙකක් තියලා තියෙනවා නම් මේ වගේ definition එකට. So the definition of metabolism you can write in one rule, right? The simple one, the sum of the reaction thing. They have given two rules. So what are they expecting there? They're expecting something additional, right? Other than the definition. So what's the additional thing that you can say? It includes anabolic and catabolic reaction. That also you have to write. Okay, that also you have to write. It includes anabolic reactions and catabolic reactions. Okay. Okay, state which type of adaptation the following characteristic organism is. Production of melanin highly in the skin when exposed to the sun. Right? අවුට ගියාම කළු වෙන. Not අපි නිකන් අර sunburn නෙවෙයි. This is not sunburn. Sunburn is a damage to the skin. Right? නිකන් beach එකටල sunscreen ගන්න නැතුව හිටියොත් එහෙම නැත්නම් swimming pool එකේ දවල් 12ට හිටියොත් you get red, right? I get red, right? I don't know about other people, right? So that's a damage, right? Sunlight again, your skin cells are damaged, burnt. That's not what we are talking about here, right? If you are chronically exposed, take someone who is sweeping the road, a laborer who's working outside, right? Maybe they were born with dark skin, but when they are chronically or for a long time exposed to a certain thing, there can be long-term changes, right? What is that called? It's given, right? What is it? It comes under adaptation, right? So when you are exposed to certain that kind of sort of a stimulus, right? What's the purpose of producing this melanin again and again? Why? What is the purpose of melanin? It actually protects your skin, right? It's a pigment. It absorbs the excess sunlight, right? Then protects your skin from being burnt, right? So in our tropical countries, that's why we have sort of a dark skin tone. So to protect our skin cells, because the melanin pigment will absorb the excess sunlight, right? Okay. So what are they asking? Type of adaptation. What do you mean by adaptation? Is it? What's the answer you got? Yes. So the answer I was expecting is actually adaptation, right? So it's what sort of what change is happening due to being exposed to sunlight. So the answer I was expecting is adaptation. Okay, right. The answer is adaptation. So there's a small issue there. Okay. Anyway, continue. Moving on. Name two polysaccharides found in both fungi and animals. Fungi and animals. Yes. Polysaccharide, glycogen. Right? It's the storage polysaccharide. No? Got it? Name a physical agent that affects the denaturation of proteins. Right? Right? Protein denaturation, we'll talk. Okay. Took a long time to discuss today. Let's move on. Online students, any questions? All right. Can you hear and see me clearly? Okay. No issues. Right. So today, let's move on to lipids. Neither. Take out your tubes. Right. Let's move on to lipids. I'll discuss the what I say. Okay. I'll discuss the what I say. You didn't get the tubes. Okay. So lipids. Hmm. Lipids are sort of considered unique from other types of organic molecules that we are going to talk about, right? Because mainly the biggest difference with lipids is we do not consider them as macromolecules or biopolymers. That's a big difference, right? So they are a very diverse group, right? We actually can't classify them very nicely as we do carbohydrates. ලස්සනට අපිට monosaccharides, disaccharides, polysaccharides එහෙම classify කරන්න අමාරුයි because it's diverse. So we just have some groups, right? We have a phospholipid group, we have a steroid group, like that. We have certain groups, but those are not proper classifications based on anything, right? They are diverse. They are very diverse. So that's one of the things you need to know. And the hydrophobic nature. What do you mean by hydrophobic? Highlight the word hydrophobic. What do you mean by hydrophobic? They don't like water. They are afraid of water, right? What do you mean by a phobia? Afraid of something, right? So, yeah, lipids are afraid of water. That's one of the biggest things, right? And so, because of that, can you transport lipids very easily? For example, take this lactose or sucrose. It can easily dissolve in water, and you can take it anywhere. Even glucose, right? Glucose in our blood will dissolve. It can go anywhere, right? But lipids are not water soluble. It is actually afraid of water. So what we have to do is to transport lipids. We have to package it. ලස්සනට box එකක වගේ දාලා යවන්න ඕනේ. So we'll come across that. Your lipids are actually transported surrounded by proteins. Okay, මෙහෙම protein බෝලයක් ඇතුලේ දාලා තමයි අපි lipids transport කරන්නේ. Right? Because of why? Outside you have water. Outside is aqueous, but the lipid inside is afraid of water. So, in order, if we want to transport lipids, we have to sort of pack it into a water soluble structure. So, the outside is proteins. Proteins are water soluble, but the lipids packed inside will be transported nicely like this. Okay, that's how lipids travel in blood. Keep that in mind. Okay, right. Moving on. So, lipids are diverse hydrophobic. They are large. They can be large, but they are not considered as biopolymers. Yes, polymers or macromolecules. Highlight that. They are not considered as polymers or macromolecules. Okay. Consist of carbon, hydrogen, oxygen, and the H2O ratio is not 2 to 1. Right? Not 2 to 1. How is that important? In carbohydrates, we had a fixed ratio, right? That is the ratio of water, 2 to 1. But here, our hydrogen content is much higher. Right? Hydrogen content is much higher. Highlight that. So the elemental composition is almost same, carbon, hydrogen, oxygen, but the hydrogen to oxygen ratio is not the same. Okay. Biologically important types of lipids in your syllabus, we are going to learn about fats, phospholipids, and steroids. So actually nothing about steroids will be discussed, right? In your resource, there's nothing about steroids. But about fats and phospholipids, we'll discuss a lot in detail. Okay. So read, fats are made up of glycerol and fatty acids. Glycerol belongs to the alcohol group having three carbons where each of them bears single hydroxyl groups. Okay, right. Let's talk about glycerol. You learn about this in organic chemistry when you are doing alcohols, right? So the basic feature of an alcohol is that they have OH groups, right? So this glycerol is it actually has three alcohol groups like this. Okay. This is glycerol. Okay. Glycerol. What are the other components? So actually, your fats are composed of this guy, that is the glycerol. And who is the other one? That is your fatty acids. Fatty acids is a very common term that you have heard, right? Fatty acids. So fatty acids composed of, there's a carbon again, there's a double bond, right? So what is it? This group, like just for fun, what is this group? It's a ketone. Sorry, this is aldehyde. Okay, there's a hydrogen here. This is aldehyde. But here we don't consider it as a aldehyde. We are going to consider it as a fatty acid because of this special property. It has a massive carbon chain like this. This can extend up to 16 to 18 carbons, right? This carbon chain can extend out to 16 to 18 carbons. Okay. So, because මට මම දැන් මෙතනත් ඇන්ද කිය about 7. So, board එක මදි වෙනවා ඒ ටික අදින්න. So, there's a way that we are drawing these fatty acids is by like this. We are just going to draw the chain like this, like a wave, right? Like this. So this one of these joins indicate what? It indicates a carbon and a hydrogen connect. Two hydrogens connect. Okay. That's one of these bends. One of these bends indicate that. So I'll erase that. So that's how we are going to draw this kind of very long what? Hydrocarbon chains. We consider this a hydrocarbon chain. Okay. So to make a fat molecule, we need three fatty acids like this. Three fatty acids like this. Okay. Another one. Shapely? Right. Three fatty acids. Okay. So those are the ingredients. How are we going to make a fat? We are going to combine these two. What is the combination? What is the combination? Sorry. Your this has a OH, right? It's not a hydrogen here, it's a OH. Okay. Got it? So how are we going to combine? Anyway, we have to release a water, right? It's a triple dehydration reaction. We have to release three water molecules. So what do you need to make a water molecule? What do you need to make a water molecule? You need an OH and a hydrogen, right? Those are the two parts you need. Someone has to give an OH, someone has to give the hydrogen. So who is going to give the OH and who is going to give the hydrogen? That actually depends on then again who is the more acidic. That you will understand. So this is an alcohol, right? This is an alcohol. This is actually called a carboxylic acid. I sort of this is a carboxylic acid head, right? So actually in organic chemistry, you will see that actually carboxylic acids are more acidic. So this OH is going to come from this guy, like this. Again here and again here. Got it? Okay, ne? Right. Then let's make the actual fat. How does the actual fat look like? This is called a esterification reaction. Esterification. Esterification reaction. So what are the things? We have the glycerol, right? We have the glycerol, and there's a O. Okay. Hydrogen, hydrogen. Right. There's a special bond here. So in polysaccharides, we had a bond. So when two monosaccharides get together to form a disaccharide, what is the bond for? Glycosidic bond. Here we have another bond unique to your lipids. Anyone? That bond, this bond that was formed here, it's an ester bond, right? It's called an ester bond, right? So here, the two OH group is gone, right? So directly connected to the carbon, you have the ester bond, then you have the hydrocarbon chain, Okay, got it. Any doubts? So this is a sorry, this is a esterification reaction. Esterification reaction. These are your 3 times fatty acids, 3 fatty acids, 3x fatty acids. We don't actually call this as a fat in the sort of the scientific way. What is the sort of the proper name of this? Triacyl glycerol is the name. Okay, triacyl glycerol. Okay, draw this in the space that you have in the tube. Draw this in the space. There's a space, right, in the tube. Yeah. Ah, no, H2O released. Very good. Where do we write? Method, right? 3 waters go out. Balance. Yeah, thank you. Right. Online students, draw this. There's a space here, right? Under fats. There's a space, right? Online students, can you see in the tube? There's a space here. So I actually have exams these days. So a bit it's stressed out the exams. It's not exaggerating, almost the A level, like the entire A level syllabus is going to be questioned in that exam. Okay. So we have 5 subjects: pharmacology, parasitology, microbiology, community medicine, and pathology. Okay. So it's like chemistry, physics, a little bit of physics you need in pathology. So it's very stressful. People get repeated, right? People in medicine, if you get repeated more than 2 times, you will be put down to the below batch. පල්ලෙහා batch එකට දානවා. Right? That's like grade 12 වල final term test එක fail වුණොත් ආපහු 12 වසරේ මා ඉන්න වෙනවා වගේ. Right? So in your third year, if you get repeated, ආපහු second year යන්න වෙනවා. So in medical faculty, you have to spend 6 years no? So එතකොට අවුරුදු 7ක් විතර ඒක අසරලා ඉන්නේ. Be very careful about who gives the OH. Okay. When you are drawing this, be very careful, mindful about who is giving the OH. Okay. Who is giving the OH? That is very important. and label the red bond. What is this bond? Yes, ester bond. This is a carboxylic acid. This head is a carboxylic acid group. Okay. This part, this part is a carboxylic acid group. Okay. This is the alcohol, and you will learn in chemistry. Carboxylic acid is more acidic than the alcohol. So, who is going to give the OH? It's going to be the carboxylic acid. Yes, I know acids are supposed to give hydrogen. Okay. Don't ask me how, but in the carboxylic acid case, it acidity actually gives the OH, right? Acidic when the OH check then. Okay. So the big, more acidic one is going to give the OH. I can't remember why, but in organic chemistry you will learn, right? So carboxylic acid are actually the most acidic organic molecules. I think then you have the after carboxylic acids you have phenols. I think then on homothiathia. Online students drawing, right? Okay. Any issues? You can see the full extension of the board, right? Yeah, I forgot to not to go that far, but okay, done. Yeah, betul. Okay. Okay, moving on. So they say fatty acids are hydrocarbon chains with a long, usually 6 to 18. So this number is very variable. Okay, the number of hydrocarbons is very variable. They say 6 to 18, but actually there are much more longer ones than that. Okay, with a carboxyl group at its one terminal. So I told you about this. Then fatty acid molecules bind to each hydroxyl group of the glycerol by an ester bond. The resulting fat molecules are called triacyl glycerols or triglycerides. Okay. Right. Hydrocarbon chains of fatty acids contribute to the hydrophobic nature of the fats. So this tail doesn't like water. This tail, we call it the hydrocarbon chain. It doesn't like water, right? So that's what actually dominates this. Because can you see? Like there's only one, two carbon atoms, right? There's only two carbon atoms here. But here, up to here, there's about 6 to 18 carbon atoms. So this is the bigger part of the molecule. So this hydrocarbon chain is going to define how this entire thing behaves. So how does it behave? It behaves hydrophobic, right? It doesn't like water. Because why? The tail doesn't like water, right? So netting satawa control karනවා, right? So if the tails don't like water, then the entire molecule doesn't like water. Okay, right. So if they ask what part of the fat or the triacyl glycerol contributes to the hydrophobic nature, the answer should be what? Hydrocarbon tails, right? Then we come to a classification of fatty acids. A classification of fatty acids. So based, I told you, right? Natta tamai satawa decide karane. So according to properties of this fatty acid or the hydrocarbon chain, there are two types of fatty acids. There are two types of fatty acids. Based on the two types of fatty acids, you have two types of entire fats, right? Fatty acids decide what kind of a fat it is, right? So fatty acids we have Uta as, yes, saturated and unsaturated. Saturated and unsaturated, right? The saturated fatty acid has nicely, all the hydrocarbon chains are nicely connected by single bonds like this. Like this, right? ලස්සනට කට්ටියම single bonds වලින් connect වෙලා තියෙනවා. Okay. That is a saturated, right? What do you mean by saturated? That something is full, right? Saturated. So this chain, can you see? It's full of hydrogen, right? Nothing, no hydrogen is missing. So this is saturated. What about unsaturated? Unsaturated, I'll draw here because we have to talk a little bit more about unsaturated, right? So unsaturated, there's actually two types of unsaturated. You don't, yeah, it's mentioned, right? So you have two types of unsaturated as cis and trans. You have heard like cisgender people and transgender people, right? That's sort of a very common discussion that we have, right? So cis means whatever the cis or trans. Unsaturated means somewhere in this hydrocarbon chain you have double bonds there, right? So what happens when you have double bonds? How many bonds can a single carbon form? Four, right? Or chemistry, right? Why can it only form four covalent bonds? It has four electrons in the outer shell or something, right? Electrons හතරයිනේ තියෙන්නේ. So covalent bonds හතරයිනේ හදන්න පුළුවන්. Right? ඕල වල තිබ්බත් නේද? So the maximum number of bonds the carbon can form is four, right? But what happens when it makes a double bond? A double bond so between the two carbons there's a double bond. So මෙතන දෙකක් තියෙනවා already. Those are covalent bonds. So how many other bonds can it form? It can form one here, that is one hydrogen, and there's another one has to be for what? The in the carbon they join in it, right? So we'll draw it in other color. Right? So a double bond if they make a double bond, that limits the number of hydrogens that they can bind. They can only bind to what? One hydrogen each. Similarly, this is again very similar to it's a kind of unsaturated, but the difference is the two hydrogens are on the same side. Trans it's on the two opposite sides. Cis on the same side. Okay, got it? Online students. So get it into your heads, right? If a triacyl glycerol is composed of only saturated fatty acids, it's what? Yes, it's a saturated fat, right? It's a saturated fat. If it has a single unsaturated fatty acid, it becomes an unsaturated fat. Unsaturated fatty acids are of two types, as cis and trans, right? What makes them unsaturated? The double bond. Easy enough, right? So read, saturated fats are made up of saturated fatty acids with hydrocarbon chains having no double bonds. Please highlight that. Under saturated fats, please highlight made of saturated fatty acids with hydrocarbon chains having no double bonds. So they can ask you what is a saturated fat. If they ask you what is a saturated fat, this is the answer you have to give. Okay. Usually animal fats come under this category. They are mostly solid at room temperature. Butter. So butter දිය වෙනවනේ කියලා එතකොට කියයි. Butter එළිය තිබ්බොත් දිය වෙනවනේ. ඇයි ඒ butter දිය වෙන්නේ? එතකොට ඒවා solid කියලා කියවන. Yeah. What is your room temperature? We are not even in the room temperature at this moment. Scientific room temperature. What is the scientific room temperature? Have you heard? What is the scientific like 25, no? 25 degrees C is the sort of what we consider as the room temperature. But what is our room temperature? අපි ඉන්න ලංකාවේ room temperature කීය සාමාන්‍ය? සාමාන්‍ය දවල්ට 33ට 4ට යනවානේ, right? මේ දවල්ේ අද නම් හොඳයි. අද maybe the room temperature might be around 30 because it's a rainy day, right? But what's our room temperature? Is it the proper one? No. So in Sri Lanka, yeah, butter diya, right? In Sri Lankan room temperature, butter dissolves, but butter, proper quality butter, right, composed of only saturated fats should not dissolve, right? It should remain as a solid. Okay, that's what it says. Then unsaturated fats, Huda, highlight the definition, made up of unsaturated fatty acids with hydrocarbon chains having one or double bonds, right? So inside this chain, there can be one, sorry, there can be one double bond or there can be more than that. That depends. Okay, that depends. That depends. Okay. So usually plant fats come under this category, right? Plant oils, we say, uh, are usually in the liquid state because they have a lot of unsaturated fatty acids, making them what? Unsaturated fats. Vegetable oils, right? Vegetable oils are usually liquid, no? Right. Then unsaturated fats are classified based on the nature of their double bonds. Cis unsaturated fats and trans unsaturated fat. Draw this, Puta, right? In your tube. In the next page, I have given you uh, unsaturated fats are classified based on the nature of their double bonds, right? So under that, I have cis and trans, right? Given. So they are in this space. There's a space, right? Draw the two kinds. Cis on the same side, trans on the opposite sides. Online students, can you see? There's a space here besides the two uh, fats. Can you see? Double bond එකක් තිබ්බ ගමන් මොකද වෙන්නේ? It will cause bending. එතනින් නැවෙනවා. Right? එතනින් නැවිච්ච ගමන් මොකද වෙන්නේ? It affects the packing, right? If it's bent, it affects the packing, right? එතකොට ලස්සනට pack වෙන්න බෑ. It can't stay as a solid. So if there is a bending because of a double bond, it is going to likely cause it to be a liquid, right? ලස්සනට pack වෙන්න බෑ because there's a bend. But if it's a saturated fat, it can nicely pack and become a solid, like what? Animal fats, butter. Okay. Okay. Right. Consumption of excess saturated fats and what do they say? Trans unsaturated fats contribute to atherosclerosis. Please correct that. That's from your, the name is not properly spelled right. It is athero. Okay. Not pro. If you check your resource book, that's also wrong. So I didn't type this. So that person has directly copied this. This is called, the condition is called atherosclerosis. Athero. Atherosclerosis, I can talk about a whole day about it, right? Atherosclerosis. Please correct it. It's wrong, right? Spelling. So after interval, I'll show you a little bit about, I'll talk a little bit about atherosclerosis. For now, let's move on. Okay. Right. Let's come to phospholipids with the projector. I'll show you. Okay. Let's move on to phospholipids. Any questions about this? You have to be able to draw this. Okay. You have to be able to draw the esterification procedure, right? Keep in mind, if I ask you to draw something in the class, that is going to be question for you to draw in the paper, right? Board it's likely I'll ask you to draw it, then it can be asked from you in the paper, right? This you have to be able to draw. මට වගේ කොයි එකද මේ බොන්ඩ් වෙන්නේ මොන OH එක කවුද දෙන්නේ කියලා අමතක වෙන්න බෑ, right? Okay. Got it. So that's about triacyl glycerols. They are very common. They are everywhere in the nature, right? No issue. Moving on to phospholipids. Phospholipids is again important group of lipids, right? Important group. They have a very similar structure to your triacyl glycerols. Have you heard where do you have phospholipids? Phospholipids koi dinne. They play such an important role as the boundary of all the living cells. Any cell in the world has a plasma membrane. Have you heard the plasma membrane? Cell membrane surrounding a cell, you have a membrane, right? That is the outer boundary, right? Cell like outer boundary there's a plasma membrane. So that plasma membrane is actually composed of this phospholipid. Okay, phospholipid. What is the composition? You don't need to actually be able to draw a phospholipid, right? Phospholipid it's given, right? No need to memorize the structure. In the tute, I haven't given, but in the resource, it says no need to memorize the structure, right? You don't need to be able to draw a phospholipid. Got it? Right. Can you see? So therefore, I'm not going to draw it, right? It's going to take time. You can draw a phospholipid very simply like this. It has a head, okay, and it has two tails. Phospholipid. Okay, this is a head. This head likes water. It's a likes water now. It's a hydrophilic head. Hydrophilic head. What are the components of this hydrophilic head? There are several, right? These two are what? These are the two people we already know. These are hydrocarbon chains. These are what we call as. So, what is the nature of hydrocarbon? They don't like water, eh? By any other, right? So, these are yes, phobic tails. Hydrophobic tails. What are the components that are included in this head? Right. So, Buddha, actually, if we sort of draw this a little bit closer, here inside you have the two. Can you remember? This is the carboxylic group that is sort of included in the head, right? So, from here downwards, you have the hydrocarbon chain, right? Right. Look carefully in the diagram in the resource, in the tube. Look carefully. Can you see? This is the limit. So, this carboxyl group is included inside the head. Can you see? Of the fatty acid, what we say as the head includes this part of the fatty acid. Can you see? Only the hydrocarbon chains are here in the hydrophobic tails. Look carefully, right? So, what are the components of the head? Right? They say there's a phosphate group, right? In the head, there's a phosphate group, and then what? Attached to the phosphate, there can be additional molecules such as choline, right? It's a polar molecule, so it doesn't have to be choline, right? Any polar molecule is fine, right? There's a diversity of phospholipids as well. So the most common one is having choline. So let's have choline there. And who else? The glycerol, right? You know, our friend, there's a glycerol. And finally, our carboxyl acid group, part of the fatty acid is also included in the head, right? So, carboxyl part of the fatty acid. You get it? This is very important. What are the components of the hydrophilic head? We have four parts. take this down. Online students, any questions? Yes. Hydrophobic tail is composed of what? The hydrocarbon chains. Yes, hydrocarbon chains. No, these are small theory points, right? The paper weight is uh, actually very low, right? So I'm not going to spend much time in those things, right? I can give you like tutes and papers for you to fill in the blanks. අන්තිමට කොළ ගොඩක් එකතු වෙයි. But at the end of the day, everything is in the resource, right? So what matters is within these 4 hours, just understand nicely, right? Understand nicely, go home and study the resource, right? අවුරුදු දෙකට පස්සේ මෙච්චර කොළ ගොඩක් තියෙයි tutes කියලා. But do you need to study those? No. What do you need to study? The resource. Okay, so next week I'm going to be doing a small seminar that is on microscopes, right? I'll try to bring a microscope and let's do the practical, that is observing a onion peel, right? Can't sure I have to steal from my lab, right? අර හොරෙන් අර පැත්තකට දාලා තියෙන bit අර මාට්ටු වෙන්නේ නැති එකක් තමයි ගේන්න වෙන්නේ, right? Let's see, right? At least you will be able to see the parts of the microscope if it doesn't work. Okay, clear. So this is the phospholipid, right? In that space, do we have space? No need, right? By looking at this, can you tell me what are the differences between this phospholipid and the triacyl glycerol we talked about earlier? What are the differences that you can see? Yeah, there's a hydrophilic head. Yes. Structurally, how many what fatty acid groups are in fatty acid chain? There's only two. In phospholipids, there's only two fatty acids, right? There's only two hydrocarbon chains. That means only two fatty acids are involved in forming a phospholipid. In triacyl glycerol, how many? Three, right? That's the biggest difference that you need to know. Okay. So actually, in triacyl glycerol, there's a small hydrophilic head, right? There's a small hydrophilic head in the previous molecule that we discussed, but that is completely dominated by this, the hydrophobic tail. So there's not even a chance for it to be a small hydrophilic part. But here, what are the difference? එක hydrophobic කාරයක් නැහැ. So එයාගේ hydrophobic effect එක අඩු වෙනවා. මොකද දෙන්නයි ඉන්නේ. Right? එක hydrophobic තොර hydrophobic side drops a little bit. Additionally, there's a polar molecule such as choline and a phosphate group. So these two things will increase the hydrophilic nature compared to normal fat. Right? Compared to normal fat, it will increase the hydrophilic nature, these two things, and lacking one minus hydrocarbon chain is going to reduce the hydrophobic nature. Get it? Right? එතකොට මහා ලොකු hydrophobic, ඒ කියන්නේ අරය තරම් hydrophobic නැහැ. But you can sort of differentiate. අපි fat එකක් ගත්තොත් it will be dominated by the hydrophobic nature because it has three fatty acids and the sort of the hydrophilic parts are made in විතරයි. So they are sort of completely dominated by the hydrocarbon chains. But here in a phospholipid එයාගොල්ලන්ගේ උදව්වට to increase the hydrophilic nature, you have the phosphate group and the additional polar molecule. So these two people are going to increase the hydrophilic nature a bit. So the result is you get a mixture of hydrophobic and hydrophilic. This is a special molecule having both hydrophilic and hydrophobic nature in the same molecule. What do you know? Do you know what it is called? Having both hydrophilic and hydrophobic nature in the same molecule. It's called amphipathic. Amphipathic. Right? එක පැත්තක් වතුරට කැමති, එක පැත්තක් වතුරට කැමති. Right? Amphipathic. Write down there in the tube. Write down there. There's a space, right? On the side of the phospholipid. Yes. Nathaniel? Write down. Having both hydrophilic, write down. Having both hydrophilic. Online students, write down what is amphipathic. Okay. The word is in the board. Having both hydrophilic and hydrophobic nature. Having both hydrophilic and hydrophobic nature in the same molecule is called amphipathic. Is called amphipathic nature. Is called amphipathic nature. Amphipathic කියන එක තේරුණා නේද? So this amphipathic nature is very, very important when we are studying about the cell membrane, right? It's going to, I'll tell you right now what's the importance, right? අපි when මේ cell membrane කියන එක්කෙනාව ඇන්දොත් it's going to look like this, right? So outside the cell, what do you have? Outside the cell you have water. Inside the cell also you have water. ඇතුළෙත් වතුර, එළියෙත් වතුර. You see the errors. Right. So inside a cell you have water, outside a cell also you have water. So how can they orientate? Right? Only the head likes water. Tails don't like water. How are these phospholipids going to create our plasma membrane? So you have to be able to understand that they have to orientate themselves in a special way. Right? එක layer එකක් තියෙනවා. So එළිය කවුද ඉන්නේ? Water. Think this is outside the cell. This is outside. Vatur. Aqueous environment. Right? Okay. Inside the cell, there is also water. What do you have inside? What we call as a cytoplasm. Have you heard? Cytoplasm. We talked about water last week. Biological media. Inside the cell, you have water. Right? So, can you leave it like this? No, because the tails don't like water. Yeah. So, someone has to protect the tails from the water. That is done by another layer. Another layer of phospholipids. Their heads are oriented towards the water inside. So, their tails are to the tails of these people. නැටි නැටි එකට connect කරන්නේ. The tails are facing each other. So this is what we call as a hydrophobic interior, right? The hydrophilic heads are facing inside and outside. This is the nice arrangement or orientation of how phospholipids form the plasma membrane. Draw this. Quickly draw this there. There's a space. Yeah, here under the functions of lipids, two tip method draw a small plasma membrane like this. Label inside, outside, and label this as. Give me a nice name. So these are phospholipids. Is it a single layer? How many layers? Two layers. So namak kya? So these are phospholipids arranged as two layers. ඔයගලන්ට scientific නම් දෙන්න පුළුවන්. A bilayer, right? This is a phospholipid bilayer. Phospholipid bilayer. Bi means doesn't like both men and girls කියන එක නෙවෙයි, right? It has two layers. Bilayer. දැන් මේකටත් phospholipid කියන එකටත් ඕන මොන්ටිසෝරි ළමයට නම දෙන්න පුළුවන්. It's a lipid with a phosphate group. It's a phospholipid, right? It's like that. So this is how the plasma membrane is formed by phospholipids. Okay? නිදිමතද නෑනේ? වැස්ස කියලා නිදිමතක් නෑනේ. Okay. Right. Then let's a few words about steroids. A few words about steroids. What comes to your head when you hear steroids? Steroids කිව්වම මොකක්ද ඔලුවට? Like steroid ගහනවා කියන එක නේද? Sorry, steroid abuse කියන එක, right? In sports, especially like bodybuilding, right? Steroids use කරනවා කියලා තියෙනවානේ, and in any sport, right? Those are called what? Those are called anabolic steroids. Have you heard? If you take bodybuilding, so I can talk about that for a long time, right? In bodybuilding, what we use, not we, people use is different versions of synthetic versions of the male hormone. What is the male hormone? Testosterone, right? So testosterone is a steroid hormone, right? It's a steroid hormone, right? So these steroid hormones and other things in our body are synthesized from a very simple basic steroid, right? අපි නිකන් හැමදේම හදන්න ගන්න එක universal steroid එකක් තියෙනවා. That is your cholesterol. Cholesterol can be used to make almost all the steroids that you have in your body, right? Usually sex hormones like your testosterone and also the female estrogen, progesterone are steroid hormones. So they are made using cholesterol. So we call cholesterol as a precursor. Right? අපි cholesterol අරගෙන යාව එක එකව කරලා testosterone හදනවා. testosterone හදන්නේ කවුද? අපේ ඇඟේ කවුද? The testes, right? In the female, estrogen හදන්නේ ovaries, right? So to that organ, what do we supply? We supply cholesterol. How do we get cholesterol? From the diet, right? From our diet, and also in our body, in the liver, we can make cholesterol, right? When කන lipids වලින් cholesterol හදන්න පුළුවන්. ඒකයි අර කියන්නේ if you are like genetically predisposed to have high cholesterol levels, ඔයගොල්ලෝ cholesterol කොච්චර නොකා හිටියත් ඔයගොල්ලෝ liver එකෙන් cholesterol හදනවා. Right? If you are genetically is sort of having a risk of having high lipid levels, however careful you are going to get high cholesterol, right? එතකොට බෙහෙත් බොන්න වෙනවා. Right? My point is, cholesterol is the precursor steroid. You can see the structure of cholesterol in the tube. Can you see this? It's a it has 4 rings, right? That's a very unique feature of cholesterol. It has 4 rings, right? That is the sort of the unique feature of a steroid, right? Steroids you don't need to know, just that is enough. Any question? All right. I think in small letters it's given, right? Cholesterol is the molecule from which other steroids, including sex hormones, are synthesized, right? Steroids vary in the chemical groups attached to their four interconnected rings. ඔය ටික දැනගත්තා නම් ඇති. චුටියට තියෙනවනේ diagram එකේ. ඔය ටික දැනගත්තා නම් ඇති. Right? Steroid hormones. Okay. Any questions? Let's move on to functions of lipids. Okay. Food reserve as an energy source. Triacylglycerols as fats and oils. Right. If you take in our bodies, where do we have our fat storage? Here. There's a fat storage. Right. There are places in our body, sort of in our arms, around your thighs, main mennang badavateta. Right. There's a fat storage. Right. Where what do you store there? Mainly triacyl glycerols. We are animals, ne? So, අපේ ඇඟේ තියෙන triacyl glycerols. But that triacyl glycerols is actually converted to, I mean, it is actually stored in kind of a tissue called adipose. Adipose. I'll show you some adipose. The cells are sort of filled with fat. Fat cells වලින් හැදිලා. Right? So if you like in a dead body, so when we are in our first 2 years, we have to cut real human bodies. So if it's a very sort of a fat person කියන්න අමාරුයි. Obese person කෙනෙක් නම් හරි අමාරුයි structures බලන්න, because why? Everything is covered by fat. Right? We have to scrape fat from everywhere. Right? For example, if you want to look at some nerves in the chest or the abdomen, nerves are blood supply, it's very difficult because everything is surrounded and hidden by the fat. කෙට්ටු මිනිස්සෙක් කෙට්ටු body එකක් අම්මුනොත් they are very happy. Right? Because all the structures are nicely visible. Right? That is because if you eat a lot of food, whether it's carbohydrates or fats, that excessive energy will be converted to what? Triacylglycerol and will be stored in your adipose tissue. Okay. That adipose tissue is everywhere, but most commonly you can see around the abdomen, legs, and sometimes neck, right? In arms, fat right? So those are the places usually you have your fat storage. Right? In plants, you have oils as the storage, right? That is clear, right? Maintain the fluidity of the plasma membrane by phospholipids and cholesterol, right? So actually, this we will discuss in detail when we are doing the plasma membrane. These phospholipids are not tightly connected to each other. So they are free to move wherever. I mean, they are like nicely interconnected, like nicely packed, but they are not fixed. So they can move here and there. And also, these cholesterol molecules are sort of found here and there like this. In between phospholipids, you have some cholesterol. This cholesterol actually again contributes to keep this nicely fluid, nicely tightly packed and keep it like a nice fluidic nature. This random cholesterol molecules also contribute. So in your diagram that you drew, draw some cholesterol molecules. Draw some cholesterol molecules. Online students, නිදි නෑ නේද වැස්ස කියලා. ගෙදර හිටියා නම් හැබැයි මම නම් නිදාගත්තා. right? Okay. So that's what they mean by phospholipids and cholesterol contributing to maintain the fluidity. Got it, right? Then act as signaling molecules, hormones that travel through the body. Right? Based on using cholesterol, you can make this kind of steroid hormones. Right? These steroid hormones are signals. When they reach a certain place, they are going to give a signal. So when you get testosterone, what is going to be the signal? If testosterone goes to your muscles, what is going to be the signal? Bodybuilders testosterone inject කරන්නේ මේකට for the muscle to get big, because that's a male feature, right? Having big muscles is a male feature, right? So the testosterone message is what? To increase male characteristics. So that is what we call as a signaling molecule. Get it? Right? And then similarly, after puberty, so there are female hormones going to increase, right? That is going to increase, start ovulation and other female characteristics, right? So those are different signals. Those signals are taken by hormones, right? Found as component of cell membranes. Again, that's the same thing, no? Phospholipids and cholesterol. Okay, clear? Right. At the end of this section, there are some questions. Right. Come to this stage. After nucleic acids also, online students. So we were right here. So after proteins and nucleic acids, we have our questions. Neither. So in your questions, Buddha, can you see this MCQ? Which of the following statements regarding lipids is correct? Yeah. Can you see? Which of the following statements regarding lipids is correct? Please do that MCQ. 30 seconds. 30 seconds. Quickly. Online students, can you see? Send me the answer, please. It says number 1. I didn't change the question numbers because, can you see? So that is the usual place. The first MCQ can be sometimes from your chemical basis. Okay. Online students, send me the answers. I want more interaction from the online people. Got the answer? So you can watch the previous recordings and complete the tune. Okay. Huh? Yeah. I'll. So you're in the group, right? The Yeah, you're in the group. I'll put the recordings again. Right. So this is temporary, right? So I don't usually include everything that is in the resource in the tune, right? That's useless, no. But in the first few lessons, I have included because you have to. Some students don't have the resource book yet, no? Right? But after, I mean, after I think yes, after the microscope session, there is nothing there except some diagrams and questions, right? So that's my usual tute pattern, right? No need putting the resource book again, right? So you after that you just rely on the resource. You got a resource book, right? Resource book එකක් ගත්තා? I mean NIE එකේ proper එකක් ගත්තේ නැත්තම්. Right. Resource book එකක් ගන්න. Okay. So නෑ නෑ මේ A level resource book එක. Right. ඒක ඔයගොල්ලෝ ගත්තේ කොහෙද? NIE එකේ නේ? මම අහගන්න. Okay. NIE එකේ තියෙනවා. Anyone has a resource book? Thank you. This one. Binding still okay, but so this is the resource book. This is the only grade 12 one. Right? There's another one for grade 13. So the diagram quality is a bit shit, no? For example, see, so the diagrams are not that good. So if you can, if you have the budget, get a printout of the PDFs. Okay, PDFs are there. Right? So get a printout of the PDFs, but this is fine, right? Because there's only few diagrams, no? Right? And what I sort of recommend is if you can get this bound, nice spiral bound, make our ankle, මේ කාඩ්බෝඩ් කවරය අයින් කරලා ලස්සන හાર્ඩ්බෝඩ් කවරයක් දාලා spiral bind කරලා ගත්තොත් you will be sort of in the long run you will be happy that you did, right? Okay, what about the MCQ? What's the answer? Which of the following statement regarding lipids is correct? Lipids are macromolecules consisting of carbon, hydrogen, and oxygen. Part of it is okay, part of it's wrong. Which part is wrong? Lipids are macromolecules. Lipids, even though they can be large molecules, they are not considered as macromolecules or polymers. Okay. But the second part consists of carbon, hydrogen, and oxygen. That is okay. Right in front of it, small review. Lipids are not considered as polymers. Lipids are not considered as polymers or macromolecules. Lipids are not considered as polymers or macromolecules. Online students? I didn't get the answers. Second statement, each hydrocarbon chain of saturated fat contain one double bond. Saturated now, there are no double bonds. Right? In front of it, right? In front of it, right? Saturated fats are composed of fatty acids. Saturated fats are composed of fatty acids with only single bonds. With only single bonds. With only single bonds. Okay. Those are what? Saturated fatty acids. Saturated fatty acids form saturated fats. When fats are formed, glycerol and fatty acids are joined by hydrogen bonds. Then correct it, cut the hydrogen bond and write the correct answer. Connected by what, Puda? Ester bonds. Cut the hydrogen and write the correct one. So that's a review, right? That's enough, right? Hydrogen to oxygen ratio in lipids is higher than 2:1. Yes. What do you think? That's the answer, no? Hydrogen ratio is much higher. Hydrogen, sort of, the proportion is much higher. Two phosphate groups are present in a phospholipid molecule? No, there's only one. Correct it, cut the two. One phosphate group is present. Cut the two, only one phosphate is present. What do you have two of? In phospholipid, what do you have two? We have two fatty acids. Write down. Phospholipids have two fatty acid groups. Phospholipids have two fatty acid groups. Okay. Right. Then there are some structure. Right. Can you see question number 1A? Name the three major types of lipids found in organisms. Online students, can you see? Here, in the same page, the last structure is a question number 1A. Name three major types of lipid found in organisms. What is the type of lipid that forms major component? And also, yeah, that whole question you can do. 1 minute. Try it quickly. Online students. Yeah, answer number 4 is correct, Puta. Yeah. Today you don't have any quality issues, right? Online students, you can clearly see the board, right? Give me a feedback. You can clearly see the board, right? Okay. I got a new camera. So anyone who was there from Sasanka's class? You were there. Okay. Okay, got the answers. Name the three major types of lipids found in organisms. You can say fats or triglycerols, then phospholipids and steroids. Those are the three types. What is the type of lipid that forms a major component of cell membrane? Phospholipids. We have drawn a plasma membrane as well. Okay, phospholipids. Those are easy questions. What is the main structural difference between saturated fatty acids and unsaturated fatty acids? Yes, structural difference. Yes, what do you need to write? Two rules are given. කොච්චරද දීලා තියෙනවද? How many rules? Two rules. Therefore, you need to compare, you need to write about saturated fatty acids as well as unsaturated. Right? So start with the saturated fatty acids. What do you need to write? Saturated fatty acids are made of hydrocarbon chains having no double bonds. Okay, that's the first one. Or you can say they are composed of hydrocarbon chains having only single bonds. එහෙම ලියන්නත් පුළුවන්. Right? No double bonds කියන්නත් පුළුවන්. Only single bonds කියන්නත් පුළුවන්. Right? Both is correct. Unsaturated, they're asking fatty acids, right? Yeah. So unsaturated fatty acids have hydrocarbon chains with one or more double bonds. That is very, very important. එකක් විතරක් නෙවෙයි, right? It can have more than one or more double bonds. Okay. So these are past paper questions. Can you see? At this point, those are easy, but once you have done the entire syllabus, you might forget what the hell is a unsaturated fatty acid, right? And you might forget it can have one or more double bond, right? ඒ වගේ චූටි කෑලි මිස් වෙන්න පුළුවන්, right? Okay, take a break. You can take a break, 15 minutes. After the break, we'll start proteins, huh? Okay, that's the plan. Angal proteins ටික දිවර කරමු. Online students, take a break, 15 minutes. නිදා ගන්න එපා. Different kinds of amino acids are involved in the formation of proteins. Elemental composition is carbon, hydrogen, oxygen, nitrogen, and sulfur. Sulfur is a very important point. Okay. At the center of an amino acid is an asymmetric carbon. Okay. අපි යමු how an amino acid is, what an amino acid looks like, right? So an amino acid is the building block of proteins, right? Proteins are composed of amino acids. So in order to, before studying about proteins, we need to know about the building block, right? So the amino acid we have to learn about. It's a very simple molecule, right? Basically, it has four components. At the center, there's a carbon. Okay, center there's a carbon. Connected to the carbon, we have four groups. Okay, there's a hydrogen. There's a COOH group. There's a NH2 group. This one, the other one, is called the R group. Very, very important. R group. Label this, sorry, draw this and let's label together. This is central carbon. Draw it in the space, in the tube, right? A space again, we have other things to draw there. Okay, manage your space. Central carbon. Just central carbon, right? Better say asymmetric carbon. What's this? Carboxyl group. This is the variable R group. We call it side chain. And very importantly, everyone except the side chain is known by a separate name. This may set except the R is called what? Backbone. Backbone. We have a hydrogen atom, amino group, carboxyl group, all connected to asymmetric carbon, and there's a special R group that we call as a side chain. Amino, carboxyl, carbon, hydrogen, we call it the backbone. Okay. Online students, any questions, right? Any connection issue? No, right? You can clearly see the board. So this is the basic plan of an amino acid. All right, amino acid. How many amino acids are there? How many amino acids are there? There's actually close to 100, right, in nature. But only 20 are involved in the formation of protein. That is very, very important. If there's a statement saying there's only 20 amino acids in nature, no, there is more than 20. But only 20 are involved in formation of proteins. There is a MCQ point. Okay, keep that in mind. And there's a situation. So these amino acids actually, these all 20 are different to each other. How are they different to each other? By this R group. මෙයා විතරයි වෙනස්. Everything else, carboxyl, amino, hydrogen, everything is the same In all those amino acids, what is the only difference? The R group, right? There are different R groups in the 20 different amino acids, right? So in your syllabus, at this point, we have to learn about the simplest amino acid that is called glycine, right? Simplest amino acid that is called glycine. Similar in structure, there's a COH, there's a hydrogen, there's a NH2. At the place of R, what do you have? A hydrogen. So very simple, right? This is glycine, which is one of the 20 amino acids. Draw this in on the side of the proper amino acid. Okay, this is glycine. So why do we call this the asymmetric carbon? That is because if we sort of draw a line, symmetrical line through these groups, and we sort of fold it, it sort of looks, you can't fold it on itself, right? It's not symmetrical, the two sides. Symmetrical then. But if we come to glycine, because of the two hydrogens, it is symmetrical, no? It is symmetrical. So except in glycine, in every other amino acid you have a asymmetric carbon. But in glycine you have what? A symmetric central carbon. That's and also an MCQ point. In every amino acid other than glycine, you have a asymmetric carbon. That is given there, right? In case of glycine, R is replaced by H atom. The R group, also called the side chain, differs from each amino acid, where the other groups in the backbone, other groups are in the backbone, right? What are the groups in the backbone? Carboxyl group, hydrogen group, and amino group. Wait. Clear about the structure of amino acid, you have to be able to draw this. Amino acid එකක් අඳින්න පුළුවන් වෙන්න. Okay, right. Then read further in the resource book. Amino acids have one or more carboxyl groups, may have one or more carboxyl groups and amino groups. That, what do you mean by that? What do you mean by that? At the place of the R group, you can have another carboxyl group. Ne? Carboxyl group එකක් තියෙන්න පුළුවන් R group එක. Or you can have an amino group in the R group. Right? That's what they mean. They can have more than one. එතකොට මෙතන normal carboxyl තැනක් තියෙනවා. At the side chain is also there can be either a carboxyl or amino. I'll show you some common amino acids, okay, later, the structures. The amino group has alkaline nature. Highlight. Amino group has alkaline nature. What do you mean by alkaline, Putha? It's basic, but basic not in the sense of the classical basic thing, right? In chemistry, we will talk about this Lewis acids and Lewis bases. Acids jati godak, like we can classify acids and bases on different criteria, right? So this basicity is actually due to the two lone pairs here. In nitrogen ke lone pairs දෙකක් තියෙනවා. That is because of that nature, this amino group is basic. Similar to that, this carboxyl group is acidic. Okay. Right. Then there's an important definition. When both characteristics are found in one molecule, they are known as amphoteric molecules. Highlight the word amphoteric. Earlier, we found a similar amphi කතාවක් නේද? What was that? Amphipathic කියලා කෙනෙක් හම්බුණා නේද? Both hydrophobic and hydrophilic natures in the same molecule. Amphipathic. Ne? This is here. Both acidity and basicity in the same molecule. Amphoteric. Got it? Amino acids are amphoteric is a MCQ point, right? Commonly questioned. Amino acids are amphoteric. Okay. Then hurry. Now we know about the building block. Now we are going to see how the building blocks get together. Monosaccharides, disaccharides, right? So two amino acids undergo condensation reaction by removing a water molecule. The result is a bond known as a peptide bond. Okay. The OH comes from one amino acid and the H from the other amino acid. This is called a dipeptide now. Peptides amino acids දෙකක් එකතු වෙලා හදන එක්කෙනාට අපි කියනවා dipeptide එකක් කියලා. So how is it going to work? It's like this. I'll just use these to draw it, okay? Glycine අපි connect කරමු මේ amino acid එක, right? මේ hydrogen දෙක පොඩ්ඩක් I'll draw it in a different way. One hydrogen here and like this. So who is going to give the OH? Remember this very carefully. Who is going to give the OH? The carboxyl, because it's more acidic, right? එයා තමයි OH එක දෙන්නේ. This is going to lead to form our new molecule that is called our dipeptide composed of two amino acids, right? So the dipeptide is like this, no? There's the nitrogen, NH2, and there's the carbon and the R group, right? That one, okay? That R group, and there's a hydrogen here, and there's a C double bond O, and there's a O, no, directly connected to the nitrogen, no, sorry. Yeah, no, OH is gone. There's a new bond here. Who is the new bond? Between the two amino acids, new bond? Peptide bond. Yeah, peptide bond. And so this carboxyl group is now connected to this amino group, right? So here we have nitrogen, one hydrogen. Again, now here let's draw glycine. So it's a hydrogen and hydrogen here. And again, C double one OH. This is a dipeptide. What am I forgetting? There's a water. Because why? The water was formed. So this reaction is again what? A condensation reaction. And label the bond peptide bond. Draw this. Yes. Online students, you can't see that far. No? In the space here, draw the formation of a dipeptide, right? Space given here. Take this one quickly. No need to label these again. Just draw this amino acid, draw the other amino acid, condense them, and draw the dipeptide. आपको label करने अंडे पा carboxyl में just simply do it. मत कम्मेली का मैं एक में, right? Don't forget the water at the end. It's a condensation or a dehydration reaction. My friends, the full tute is now available in the group. Okay, the full tute is now available in the group. It's a bit big because it has some diagrams. So check the group, please. The full tute is now available. It's being sent. Okay, so once you have got, please download it. By next month, we will start the delivery process of the tute. Okay, so you can have the physical copy of the tute with you. Okay, so for now, for another week, just use the soft copy PDF. Right. Okay, I'll show you some. Okay, okay, done. Delivered. Yes, so this is some of the amino acids. It's a bit. So I just wanted to show you the diversity. Completed drawing, right? Tada. So can you spot glycine there? Glycine up another. Can you see how simple it is? Right? Only a hydrogen. So are we drawing the hydrogen? So now we know proper organic ways of drawing molecules, right? Can you see? So this is the central carbon. We don't draw it, right? This is the amino group. This is the carboxyl group. This is the OH, sorry, the oxygen of the carboxyl group. This entire thing is the carboxyl group. So where's the hydrogen? Hydrogen is sort of down here, no? We don't draw it. Because why? Okay. Carbon and hydrogen we don't draw. Just the line segment is there. There is the simplest amino acid, right? So at these places, these are the central carbon. Here you have the central carbon. Here you have the central carbon, right? So that's how, yeah. So this one, alanine is a bit more complicated than glycine. It has CH3. We call it a methyl group. This one is much more complicated. It has this sort of a carbon ring. This has another, can you see? Aspartic acid. It has, this is a carboxyl group and this is a carboxyl. So now you understand what they say. It can have more than one carboxyl amino group. Yes. Mm, yeah, these are a little bit, right? There's only about 10 here, no? So about 8 missing. So this is the diversity, right? Everything changes based on the R group. R group is the diversifying factor. එයා තමයි මේ amino acid with change කරන්නේ. Everything else is the same. That is the backbone. Okay, moving on, moving on. Right. So come to the resource book pack again. So we talked about the condensation, how a dipeptide is formed, right? Protein is composed of one or more polypeptide chains which are composed of amino acids. So like this, how two amino acids connect to form a dipeptide, it goes on like that, right? Dikkata chain එකක් හදාගෙන යනවා, right? Peptide bond formation, right? That is called what? A polypeptide chain, right? So what we need at the end of the day is a protein. Proteins are found everywhere in your body, right? So when you're using a skeletal muscle, it has proteins called myosin and actin mainly, right? And in your bones, you have proteins, right? And inside your cells, most of the functions, the enzymes, right? And let's say there are storage proteins, structural proteins, so many things. If you take your skin, it is mainly composed of proteinous cells, right? So proteins are everywhere. So to have a protein that does a certain function, just the series of amino acids is not enough. We have to modify it. Aromator mix kalla, right? We have to change it in several ways in order to get our final product. That is a working protein, right? A protein with a function, right? To do that, this polypeptide chains undergoes a stepwise process, right? That is called the level of protein structure. So from level 1, that is the primary level or the primary structure, to the final level, that is called the fourth level, quaternary structure, these chains undergo some changes. Okay, let's start. There are 4 levels of protein structure which play important role in their functions, right? These are primary, secondary, tertiary, and quaternary. Okay, come to the primary structure. Please highlight, these are definition. They can ask what is the primary structure. The unique sequence of linearly arranged amino acids linked by peptide bonds is the primary structure. That's the definition. Can you see in the tube? You can see some amino acids that are connected, right? That are interconnected by peptide bonds, right? That is a polypeptide chain. Polypeptide chain level is the primary structure. Got it? Quickly moving on to the secondary structure. The primary structure, which is what? Just the chain, it coils and folds as a result of intramolecular hydrogen bonds. Very, very important. Highlight the word intramolecular. Intramolecular hydrogen bonds between the oxygen atoms of the carboxyl groups and hydrogen atoms of the amino groups of the same polypeptide chain backbone to form the secondary structure. I cannot highlight the importance of knowing the difference between intramolecular and intermolecular. What do you mean by intra? Within, right? Intra means within the same molecule. Inter means international, right? Between two nations, right? So intermolecular means what? Between two different molecules, right? You have to memorize what sort of molecules at what stage you have intramolecular bonds and intermolecular bonds. Okay, right. I had a good diagram to show this. Proteins. Yes. Ah, yes, this diagram. Can you see online students? I don't know if it's that clear. Can you see? ඔයාලට පේනවද? Right. What can you see? This is the same polypeptide chain. මෙහෙම යනවා. Right? What can you see? These are the R groups. What are the R groups? The green color. Those are the side chains. ඒගොල්ලම වැඩි හිතන්නේ නැහැ. Right? We don't care about the side chains at this point. At the secondary level, right? Secondary structure level, we don't think about them. What are we concerned about? We are concerned about this. It's not that clear. This is the carbon. Connected to the carbon, what do you have? Oxygen, right? There's an oxygen there. And here there's a nitrogen, right? That is of the amino group. There's a hydrogen of the amino group. Yeah. Whenever you have this sort of oxygens and hydrogens, what are they going to do? Because of the electronegativity difference, this hydrogen is slightly positive, this oxygen is slightly negative. Neither. Right? You know about hydrogen bonding, right? So oxygen is slightly negative, hydrogen is slightly positive. These two form hydrogen bonds. Right? You clearly have to know what are the two groups that are involved. The oxygen of the carboxyl, hydrogen of the amino of the same amino acid or the polypeptide chain gets together to do what? To form the secondary structure. Right? Intramolecular hydrogen bonding. MCQ points, right? Intramolecular hydrogen bonding. So this is one of the ways this can do. This is a helix, right? This is called the alpha helix. Okay, this is one of the forms of the secondary structure. There is another one that is in your resource as well. It's like the pages of a book, right? If you take this and if you fold it like this is now a beta pleated sheet like this, right? This is how a beta pleated sheet looks like. It's in several layers like this. Can you see? If you have folded like a piece of paper, that's how the beta pleated sheet looks like. The helix is like this, like DNA වගේ මෙහෙම helix වෙලා. There are two forms in your syllabus that you need to know. That is the beta pleated sheet that is like this, and the alpha helix. And you have to know the examples in nature. There's a protein called keratin. Where do you have keratin? Everybody should know. Keratin කොහෙද? Yeah. Hair is a very common place, and in your nails, or even in the surface of the skin, you have keratin. Right? Goda dry වුණාම සුදු පාටට කොහොම යන්නේ? Dry skin එකේ. That is keratin being like because of the nail, keratin is being like sloughed off කියලා කියන්නේ නිකන් අර සුදු පාට powder එකක් වගේ ඉන්නේ keratin, right? In dry skin. So keratin is a example for alpha helix. Highlight. Keratin is the example for alpha helix. And for the beta pleated sheet, your spider silk, they say, is in the form of this beta pleated sheet. Right? ඒක නිකන් නූලක් වගේ තමයි එන්නේ, but I don't know about that. It's supposed to be like a pleated sheet in the microscope, right? Molecular level එකේ ඒක sheet එකක් විදියට තියෙනවා. Okay. Yeah. So alpha helix. Yeah. So the beta pleated sheet is like this, right? So it forms hydrogen bonds like this. Can you see? The oxygen of the carboxyl and the hydrogen of the amino group form a beta pleated sheet like this, right? Pages folded on top of each other. That is the beta pleated sheet. There are different formations of beta pleated sheet, right? එක orientations තියෙන්න පුළුවන්. You don't need those. Okay. There are other formations such as beta bend කියලා එකක් තියෙනවා. You don't need that, right? Just remember alpha helix and beta pleated sheet. So this red color area is what? That is the alpha helix. And these areas, these are beta pleated sheets, right? Can you see? In the same protein, you can have sometimes the alpha and beta structures as well. Sometimes, right? We'll talk about this. In some proteins, such as big proteins such as hemoglobin, you can have this mixture of structures. Clear? Moving on to tertiary structure. Highlight the secondary polypeptide chain bends and folds extensively, forming, these words are very important, ඊළඟට එන words ටික, precise, compact, unique, functional 3D shape, right, resulting from the following interactions. Online students, this same diagram I have included in the tute. So can you spot where are the interactions occurring between the side chains? Can you see that, right? These new interactions are between what? The side chains. Previously, where did we had the hydrogen bonds? Was it between side chains? Where was the previous secondary structure that the hydrogen bonds between amino group and carboxyl group? Ne? So those are parts of the backbone. Side chain එක අපි ඉග්නෝර් කරා නේද? R group එක ගණන් ගන්න එපා කියලා මම කිව්වේ කොළ පාටටි. Right? But now we are ගණන් ගන්නවා, right? So now in the tertiary structure, it is formed by interactions between side chains. Then අපි backbone එක අමතක කරන්නේ. So this purple line is your backbone. Backbone එක අපි අඳින්නේ. What are the things in the backbone? There's a carbon, there's a carboxyl, there's an amino, and there's a hydrogen atom. ඔය ටික අපි දැන් අඳින්නේවත් නැහැ. We are not going to even think about it. Because at the tertiary structure level, what matters? Side chain, right? Side chain එක විතරයි අදා. Right? So there are several types of interactions that can occur between different side chains. What are the different kinds of interactions? They are given in the resource. Hydrogen bonds can occur between side chains. Okay, can you see? This is a side chain. Here there's a OH, there's a O, and between that hydrogen and this oxygen, there can be a hydrogen bond. Right? What is the other type? Disulfide bond. There's a special name. It's called disulfide bridge. Between two sulfurs, there can be a covalent bond formation. That is a disulfide bond. Okay. What's the third one? Ionic bond. What do you mean by ionic? Positively charged and negatively charged. Because of their charge difference, they are connected. No. Can you see? NH3 plus is positive, O minus is negative, so they form an ionic bond. I hope you know ionic bonds, right? Chemistry වලට. So that's the hydrophobic interactions. It's so actually hydrophobic interactions, they are not attractive forces, right? They are repulsive forces, right? When two groups such as this CH3 or set, they don't like each other, right? They both hate water. So what is going to happen actually here is they actually repel each other away from each other. But doesn't matter if it's an attraction or a repulsion, it's interaction. It's an interaction. Interaction. So hydrophobic is an interaction. This is a place of hydrophobic interaction. And van der Waals, it's very difficult to explain. Van der Waals කියන එක ඔයගොල්ලෝ chemistry වලට කියලා දෙයි. It's just the weakest type of interaction. ඒ කියන්නේ මුකුත් අයනිකුත් නෙවෙයි, covalentුත් නෙවෙයි, hydrogen bondsුත් නෙවෙයි. Nothing. Just two molecules are attracted to each other because of their sort of their size. අර නිකන් gravity වගේ දෙයකින් පොඩ්ඩක් they attract, right? One of the weakest interactions, van der Waals. We actually ignore van der Waals in most of the situations, van der Waals bonds, right? So those can also be there. Highlight those kinds of bonds. We have 5: hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interactions, van der Waals interactions. Online students, any questions? So examples for tertiary structure. So at this level, we have a nice protein at this level. We consider these to be proteins. Okay. What are the examples? Most of the enzymes, myoglobin, albumin, right? Myoglobin is a protein that is found in your muscles, right? In muscles, myoglobin stores oxygen, right? Myoglobin stores oxygen. Have you seen when you're like doing barbecues? Sometimes it feels like from the meat you have saying like blood is coming out, right? Raw, what do you say, rare meat? It's not blood, right? It's actually the myoglobin that is coming out like blood, right? Go and search steaks. The steaks are cooked. When you squeeze it, liquid and we people think it's blood. It's not blood. It's actually the myoglobin, right? Myoglobin everything. After we have killed an animal, all the blood will be drained, so blood can't be there, right? So the myoglobin will be there. Myoglobin is also very red colored, right? Because it stores oxygen. We'll talk about myoglobin in detail, right? Albumin is a plasma protein. එක නිකන් blood වල හේ යන protein එක. It's a plasma protein. You can write down under albumin. පොඩි arrow එකක් දාලා ලියන්න. It's the commonest plasma protein. Commonest plasma protein. Myoglobin වලට arrow එකක් දාලා ලියන්න. Stores oxygen. Stores oxygen. Stores oxygen in skeletal muscles. Stores oxygen in skeletal muscles. Stores oxygen in skeletal muscles. Myoglobin. Yeah, any question? No? Stores oxygen in skeletal muscles. Okay. Tertiary structure is done. Any questions? No? Quaternary structure. So quaternary structure, so up to the tertiary structure level, who did we have? We had only one polypeptide chain, which is just the same chain. Ekama chain එකම යන්නේ. But අපි quaternary structure level එකට එනකොට we are going to take two or more chains. එතකොට මෙයා වගේම තව chain එකකුයි it can have even more chains. Two or more chains are involved in the quaternary structure level. Okay, quaternary structure level. So what's the definition? Read. Aggregation of two or more polypeptide chains involved in the formation of one functional protein. The separate chains are called protein subunits, which are held together by inter and intramolecular interactions. Right? So highlight this sentence. That's the definition. Aggregation of two or more polypeptide chains involved in formation of one functional protein. That is the definition of quaternary structure. Okay. Then in a different color highlight, separate chains are called protein subunits. Right? එතකොට මේ protein එකේ in our quaternary structure there are two or more chains. එතකොට ඒ chain වලට අපි කියන්නේ දැන් chain කියන්නේ. ඒකට අපි මොකද කියන්නේ? Subunits කියලා කියන්නේ. Right? So I'll show you hemoglobin. So can you see this is the entire story we talked about no? This is what? Yes. Amino acid, amino acid, amino acid. It's given here. Okay. That is the primary structure. Okay. What are the each amino acids are connected by? What bond? Peptide bond. Okay. Peptide bond only omega help. Right. Then what's the next level? Secondary structure. Online students, can you see? Secondary structure. What is this secondary structure? Give me the name. Alpha helix එකක් නේ මෙතන තියෙන්නේ. මෙයා මොකද්ද මෙහෙම නිකන් කොහොම ගිහිල්ලා අයි බෙන්ඩ් වෙලා තියෙන්නේ. That's a beta pleated sheet. Right? That is what? Secondary structure level. Then this is the tertiary structure. Now it's a complete functional protein. මෙහෙන් interactions වලින් හොඳට fold වෙලා compact වෙලා වර තියෙන වචන ටික compact precise 3D functional ඔන්නෝ තියෙන. ඔක්කොම features ටික එතන තියෙනවා. Tertiary structure. This is the quaternary structure. These are the separate polypeptide chains. Remember venom chains, right? How many chains can you see here? How many chains? 4. Give me a name for the separate chains. මොනවද අපි chains වලට කියන නම මොකක්ද? Subunits. Those are subunits. How many subunits are there in this protein? 4 subunits. Right? Online students following, right? Can you see the projector? I want to show you a hemoglobin. It's not here. Here. Yes. Can you see? Hemoglobin is one of the sort of proteins very important in our body. You know what is the why is it so important? Hemoglobin. What do you know about hemoglobin? Transport of oxygen. It also transports carbon dioxide as well. Okay, mainly oxygen. So how many subunits does this hemoglobin have? 4. Right? They have names also. There's a alpha 1, alpha 2, beta 1, beta 2. 4 subunits are there. This is hemoglobin molecule. There's one in your resource as well, right? There's a hemoglobin. And this is the most important part. It has an iron, right? Hemoglobin has an iron Fe2 plus, right? Fe2 plus Fe2 plus 4. For each subunit, there's Fe2 plus. So what is the function of the Fe2 plus? That's where the oxygen binds. Oxygen set to the heme group, right? And there's another quaternary structure that I have included in here, collagen. Collagen, how many subunits are there in collagen? Online students, how many subunits can you see in three different colors, right? So how many subunits? Collagen has 3 subunits, right? Write down under that. Collagen, write down under that, that collagen diagram. Collagen is composed of 3 subunits. Collagen is composed of 3 subunits. Three subunits. It is found under the skin. Found under the skin. Found under the skin. We're talking about collagen. Found under the skin. Walls of blood vessels. Walls of blood vessels. Walls of blood vessels. And in the extracellular matrix, write down, we'll talk about it. In the extracellular matrix. Extracellular matrix. That is, we'll be discussing our next, that is under cellular basis. Okay. Harinil, that is the levels of protein. Any questions up to here? Yeah, Buddha? No? No questions. Right. Then we are going to talk about important property of proteins, that is they are heat sensitive. Proteins don't like heat. Okay. Most of the proteins. There are some heat resistant proteins, but most of the proteins don't like heat. Because why? They are composed of some very sensitive interactions. Say the disulfide, iron, right? Some very sensitive interactions. So providing heat means what? You are giving energy, no? Heat is a form of energy. If you give something heat, it is going to disrupt these different bonds, right? And when that sort of energy is supplied, it's going to change the protein. Usually when a protein changes, it can't be reversed back, right? I'll give you an example. So in a raw egg white, it's mainly protein or albumin, right? Albumin. So, bitra tambuwa kya? You get it solidifies, right? Volume liquid back anything. So, sudumade, it's a solid right now. So, can you turn it back to the liquid form? No, it has been irreversibly changed. Irreversibly changed, right? And if you leave some milk out, milk tika koppe එකට දාලා එළිය තියලා තිබ්බොත් ටික දවසකින් ඉන්නේ within one day it will become sort of කැට හැදෙනවා නේ, milk sort of coagulates. That is also a type of denaturation because of some bacterial action. Bacteria කිරිවල වැඩ කරලා it's going to coagulate, solidify the milk. Can you turn it back? No, because the bonds interactions in the milk have been changed. That is called denaturation. Okay, so read the definition, highlight. Denaturation of protein is the loss of specific 3D shape due to the alteration of weak chemical bonds and interactions. Okay. Alteration of weak chemical bonds and interactions. Okay. It's enough. I mean, just knowing the definition is enough at this level, right? Nothing extra is needed. Okay. Just know the definition. Right. Agents affecting denaturation, you have to know. Memorize, please. Right. High temperature and high energy radiation. Right. Bittare thambano again, you are giving high temperature. High energy radiation. If you are microwaving the egg, there is some high energy waves. Right. And also, let's say you go in the sunlight. Right. අව්වේ අර මං කිව්වා වගේ pool එකේ බහලා ඉන්නවා දවල් 12. Right, what is going to happen? Your skin becomes red and later it becomes sort of black. And what has happened? Your skin proteins have got denatured, they have got damaged because of the UV light in the sunlight. So high energy radiation is a form of agent of denaturation. Strong acid, alkaline and high concentration of salts, right? All these things can change the different interactions, right? Salts දැම්මම ionic bonds එහෙම change, right? High acidic and high basic environments, those disulfide bonds, okay, bonds, they all get influenced, right? They get changed, right? Okay, highlight strong acids, alkaline and high concentration of salts. Then heavy metals. What are some heavy metals, you know? Tin, lead, right? Mercury. Those are damaging. Why? ඔලෙවලට තිබ්බනේ heavy metals වලින් මොනවද එක industries ටික. Margarine හදන්න ගන්නවා, nickel වලින් what do you use to make? Can't remember, right? So these heavy metals are catalysts, right? අපිට ඒගොල්ලන්ගෙන් reactions promote කරන්න ගන්න පුළුවන්. When you have a heavy metal somewhere, it's going to promote a reaction, right? So write down there heavy metals act as catalyst. What's a catalyst? Heavy metals act as catalysts. ඒගොල්ලෝ නිකන් දැන් මේ වෙලාවේ වෙන්නේ නැති reaction එකකුත් කරන්න try කරනවා catalysts, right? They are going to promote certain reactions, catalysts, right? So when the protein is in contact with a heavy metal, that is going to do some reaction that is not natural. So that is going to change the nature of the protein. Heavy metals act as catalysts. Write down. Okay. Organic solvents and detergents. Organic solvents and detergents. Phenol, hypochlorite. Hospital toilet හොදන්න, ward එක හොදන්න එහෙම use කරන්නේ. ඇයි අපි ඒවා use කරන්නේ? Bacteria රමේවනේ ඉන්නේ hospital එකේ, right? So to destroy bacteria, what do you mean? What do you need to do? You have to change the bacteria proteins. එතකොට මේ වගේ හොඳ detergent එක phenol පාරක් දාලා හොදලා දැම්මහම the bacteria are going to get killed. By what? Denaturation. Bacterial proteins are denatured. Okay? hypochlorite කරලා දෙනවා අර මේ toilet හොදන්න ගන්නේ. Chlorine කියලා කියන්නේ powder එක hypochlorite. Right? That is also going to destroy proteins, bacterial proteins. Bacteria වගේම ඔය එක එක amoeba, protozoa අර මේ වගේ disease හදන්න පුළුවන් එන set එකක් ඉන්නවා. All those can be destroyed by this sort of detergents and organic solvents. Okay, even viruses where the proteins can be destroyed. Okay, next page we have some diagrams that are sort of related to the different functions. Online students, can you see? I'll put it in the projector. First one, can you see? So which of these is the protein? Whether in protein account? That is the enzyme, right? The enzyme is the protein. මේ කවුද එන්නේ? මේ දෙන්නට නමක් කියන්න මේ එන දෙන්නට. දැන් hexagonal shape එකක් and pentagonal shape එක. Probably what? Maybe a disaccharide වෙන්න ඇති නේද? Right? Disaccharide එකක් වගේ නේද? What are the disaccharides connected by what bond? Glycosidic bond. Right? Glycosidic bond එකනේ connect වෙලා තියෙන්නේ. ඒතර මේ enzyme කියන මිනිස්සයා is a protein. What has he done? He has broken the glycosidic bond. And now we have two different monosaccharides. That is a function of a protein. Right? So when you eat sucrose කෑල්ලක් කෑවද? සීනි ටිකක් කෑවා. In your mouth you have sucrase. Not in the mouth, maybe in the small intestine. Sucrase කියලා enzyme එකක් තියෙනවා. Right? Sucrase can break this bond and separate into what? Sucrose is composed of what? Glucose and check and see, fructose. Right? So that is a function of an enzyme. Enzyme is a catalytic protein. Right? Write down under that. Write down under that. Enzymatic breakdown. Enzymatic breakdown. Write down under this diagram. Enzymatic breakdown of a disaccharide to monosaccharides. Enzymatic breakdown of a disaccharide to monosaccharides. Second one, underneath, transport protein. So this one you know, right? What are these, Pudha? Megalo මොනවද? Yes, what are they? Phospholipids. The උදේ කතා, right? These are phospholipids. So who is this? The plasma membrane or the cell membrane. Right? එහෙනම් plasma membrane එකේ phospholipids හරහා substances can't really go. To do that, there are special proteins, right? That are sort of embedded. දැන් නිකන් highway එකේ තියෙන toll gate එකක් වගේ, right? Through the toll gate, only selected things can go. Motorcycles, three wheels වලට යන්න දෙන්නේ නැහැ. Right? ඒ වගේ only the things the cell requires will be allowed to go through the transport protein. Okay? Write down under that. Transmembrane protein. Transmembrane protein. Selectively transporting material. Selectively transporting material. Selectively. ඒ කියන්නේ යා කියන්නේ ඕන දේවල් විතරයි. හැම දෙයකම එන්න බෑ. Selectively transporting material into the cell. Transmembrane protein selectively transporting material into the cell. Next function, short page. Remote එකක් තියෙනවද? Do you see a remote area? Give me 1 minute. Make a short to them. Okay. Right. Hello, students. Can you hear me? Okay. Where was I? Right. Next, let's talk about. So we talked about what? Catalytic proteins, transport proteins. Okay. Then how about this one? Signaling proteins, or let's say hormones. Okay. Mm, no, but before this, about the defensive proteins. Okay. Defensive proteins. Make out this is a virus. Virus is always a bad thing, right? A virus inside your body is always a bad thing. So inside your body, we have some proteins that are involved in defense, right? Those are called antibodies. Give me another name for antibodies. Immunoglobulins, right? Immunoglobulins. Those are proteins. What they do is they surround this virus and they sort of disrupt the processes of the virus. They can sort of kill this virus. Okay, antibodies. Similarly, it can be done to a bacterium, but bacteria is usually very massive. A virus is sort of nanometer level, right? All the viruses are very, very small, nanometer level. Bacteria are usually big, micrometer level, right? So it requires a lot of antibodies to kill a bacteria. Anyway, it's going to do it, right? So these antibodies or immunoglobulins are defensive proteins. Okay. Write down under this diagram. There's a small space, no? Write down under this diagram. Immunoglobulins can immunoglobulins can neutralize foreign substances. Those are proteins, foreign substances. Okay. What are the foreign substances you can see here? There's a bacteria, there's a virus. Those are foreign, right? Okay. Next, this one is not actually given in your chart, right? As the signaling molecules. Uh, but anyway, let's write this down. This is one end of a neuron, nerve conducting, right? This is one end of a neuron, right? This is another neuron, maybe, or maybe a muscle, right? For example, I need to flex this elbow. Neuron එකක් දිගේ muscle එකට signal එක යනවා, right? So that might be this neuron. හැබැයි this is the muscle, but that neuron is not directly plugged කරලා නැහැ to the muscle. It's not directly plugged in. There's a gap. This gap is called the synaptic cleft. ඒක ඔයගලන්ට ඕනේ දැම්ම. Right? But you will learn about this synaptic cleft. එතකොට මෙතනින් මෙහෙම එන signal එක, the signal that is coming through the neuron will have to come here through a gap. Through that gap, what do we have? We have a signaling molecule. We call them, I'll write here, neurotransmitters. Have you heard? Neurotransmitters ගැන අහලා තියෙනවද? You can write down that name. අර මොලේ neurotransmitter level එක වෙනස් වෙලා කියන්නේ sometimes you get depression, right? අර dopamine levels වෙනස් වුනම you get depression කියලා අහලා තියෙනවද? Right? වයසට යනකොට Alzheimer's කියලා disease එකක් තියෙනවා. That is all due to this neurotransmitter level. Those are proteins, signaling proteins. Okay? Write down underneath. This is something additional now, right? I mean, not additional, but for your level. You will learn about this in grade 13. Okay? Neurotransmitters transmit signals through the synaptic cleft. Write this synaptic cleft. Cleft එකක් කියන්නේ මොකද? නිකන් it's a gap, no? Synaptic cleft. These are not out of syllabus, right? Synaptic cleft. Okay. Then final one. This is a contractile protein, right? This is a very magnified view of a muscle. That one. Okay. About 30 micrometers, right? This is a muscle, right? In the muscle, two types of proteins called myosin and actin get together to do the actual contracting part, right? If you magnify into this, this is the molecular structure, right? Trillions and trillions of these kind of proteins are involved in the formation of a big muscle, right? Can you see the myosin have these sort of small hands, right? Each trillions of hands get together to do the massive muscle action that you are doing. Okay, this is contractile, or we call them motor proteins. Okay, write down under there. Actin and myosin can be found in muscle tissue. Actin and myosin can be found in muscle tissue. Okay. Right. Now jump back to the proper chart that is in the previous page that summarizes everything. Right. So catalytic proteins catalyze biochemical reactions. You have two enzymes, pepsine and amylase. Okay, pepsine and amylase. Do you need some additional information? What is pepsine or right? In the syllabus. You can write down arrow එකක් දාලා pepsine is a protein digesting enzyme. Protein digesting enzyme found in the stomach. Protein digesting enzyme found in the stomach. Body. Stomach, STOMACH, stomach. Okay. Amylase it's found in saliva and small intestine. Okay, if you want, you can write down. Amylase is found in saliva, kela, and small intestine. Structural protein, that is keratin, I told you, prevent desiccation. What is desiccation? Desiccation means water loss. Okay, වේලෙන. So our skin protects us from desiccation, right? Because inside, if you remove the skin, what do we have? We have so many wet tissues, right? Our tissues are wet with blood and body fluid, everything, right? So our tissue is, sorry, our skin is our outer covering, is like our protective layer. Skin get outermost layer has a keratin layer. Okay, the outermost layer of the skin has keratin. It's like a less thick version of Malvigi corpus, right? Scales of a fish, but very soft level, right? So scales of a fish you can see separate, but we have scales, but at a very small microscopic level. Same function, right? Prevents damage, and also prevents water loss. Okay, that's what they mean by prevent desiccation. You can add something in the skin. Prevent desiccation in the skin. Collagen we already talked about, no? Underneath the skin, right? So how does collagen underneath the skin? Okay, රටිනුත් තියෙනවා collagen උත් තියෙනවා. මෙහෙම හැම ඇද්දා කියලා ගැලවිලා අතර එනවද skin කැල්ලක්? No, right? That is because of what? Underneath our skin we have a collagen network එකක් වගේ දැලක් වගේ එකක් තියෙනවා. That is providing support, right? මෙහෙම strength එකක්ද මෙහෙම හැම ඇද්දට ගැලවෙන්නේ නැහැ. Right? Because there is a collagen layer underneath. Okay. Storage of albumin, I told you, usually found in the egg white, right? You have proteins in the egg. Casein you can find in the milk. I told you, right? Casein දිනේච උනාම තමයි අර කිරි කැටි ගහෙන් නරක් වුණාම, right? Transport hemoglobin, we talked about this. Transports oxygen and carbon dioxide. Serum albumin transport fatty acids, right? As I told, it's another way of transporting. I told you previously, can you remember? Fats are transported surrounded by proteins. Serum albumin. Okay, got it. Insulin, I think might be the most commonly known hormone. Ne? Insulin ගැන දන්නේ නැති කෙනෙක් නෑනේ. It's a most widely known hormone, right? It regulates what? Blood glucose level. So insulin ගේ sort of antagonist කවුද? You can write down. Blood glucose වලින් අපි හදන්නේ කවුද ඇඟේ? Who do we make from glucose in our body, humans? What's our storage polysaccharide? Now, revision. What's our storage polysaccharide? Glycogen, right? Glucose වලින් අපි හදනවා glycogen. This is not a one-way reaction. Glycogen හැදුවම නෙවෙයි, right? We can use this glycogen again, right? This conversion of glucose, when you have too much glucose, it will be converted to glycogen. This will be like signal for this conversion is what? Insulin hormone. Insulin. When you have too much blood glucose, that will be converted to glycogen and stored where? In the liver and muscle, right? When you don't have enough glucose, badagin inno, fasting inno, right? You're fasting for a few hours. Did you see the eraser? Right? Anyway, what's going to happen? Blood glucose level come down, no? Blood glucose level comes down. So now it's the time to use the stored glycogen, right? So this glycogen will be turned back into glucose. Who is the signal for that? So I told you this is a reversible reaction. Glycogen to glucose is catalyzed, or signal for that is called the glucagon hormone. Okay, write down this. Where is it? Eraser. Cool. Where is it? Beaver. හරිද? Write down this. Did it? It's a reversible reaction. Okay, glucose to glycogen, glycogen to glucose, and got on with here we can convert. Right. Okay. So then contractile and motor proteins we talked about, myosin and actin, they're found in muscles. Defensive we talked about immunoglobulins. What's the other name for immunoglobulins? Antibodies. Okay, antibodies. Okay, let's quickly do some questions. No, we have the paper to do. 15 minutes in a plan. Okay, yeah, take out the paper. I will send the marking scheme of the vote essay. Okay, හදලා එකක් එවන්න. And I'll pass around. Please give me your number so I can create a group. And I have already created a group. I'll add everyone to that. එතකොට මට updates දෙන්න පුළුවන් ටක් ගාලා. Okay, do the questions. Paper 4. Paper 4. Online people, paper 4, please. Cool down, cool down. So please write down your WhatsApp numbers and pass it, okay? Okay, you can do the first MCQ. You can do the second MCQ. You can't do the third one. You can't do the third one. This is from a school paper. You don't know about insulin, so you can't do the third MCQ, okay? Cut it. Paper 4, third MCQ, you can't do. Yeah, don't recommend within 3 minutes. Structure that can. I'll send you the structure of insulin, okay? Insulin monoagadikil, I'll send you a photo, like an image. Then looking at that, you can do that MCQ, okay? Insulin monoagadikil මම එවන්න. Molecular diagram එක. ඒක දිහා බලාගෙන ඔයගලන්ට ඔය structure එක කරන්න පුළුවන්. Okay? We going too fast? Any feedback? The Kianatin. Are you ready? No. No. Any feedback? No. What needs to be improved? When a class will tell you, yes, brother. Any feedback? Nothing. Okay. The earlier we cover the syllabus, the better. Bio. Like අන්තිමේදී bio අතරම class එක නවත්තලා only revision එකට යන්න පුළුවන් නම් හරි. Theory class එක අන්තිම 3-4 months වල theory class එකක් නැත්තම් හොඳයි. It's going to save you time. Just the bio revision is enough. කලින් syllabus cover කළා. Yeah. Then you can focus කොහොමත් ඉතින් in the physics class for 1 year maybe you are going to drag mechanics. Right? ඊට පස්සේ අන්තිමට අල්ල ගන්නවා gravity and electrostatics, current, electricity. ටික අන්තිමට අල්ලගෙන those are question heavy topics. But then you are going to get bit stuck. ඊළඟට chemistry වලත් අන්තිමට industrial ටික කරන්නෙම නෑ සමහර class. Right? Industrial chemistry. Right? Okay, done. හරි නේද? First MCQ. Okay, select the mismatch pair of proteins and their functions. Formation of structures. Keratin. Okay. Thank you. Everyone wrote your number, right? The keratin formation of structures. Yes, it's a structural protein. I told you, right? Hair, nails, those things are composed of keratin. So, not wrong. Contractile actin. Myosin and actin are the contractile proteins, parts of muscle. Transport within man, hemoglobin. What does hemoglobin transport right in front of it? Oxygen and carbon dioxide. Stored in structures, serum albumin. Is it a storage protein? Storage serum albumin? No. Right in front of it. Serum albumin is a transport protein. Serum albumin is a transport protein that transports fatty acids. Why do we need a special protein to transport fatty acids? Because fatty acids don't like water. Right? Okay. Next statement. Stored in structures, no. So what are they trying to fool you here? මොකක් කියලාද මෙතන stored in structures කියන එකෙන් what are they trying to misguide you here with ovalbumin, right? Ovalbumin is a storage protein, right? So they are going to sort of confuse you with there's another albumin. So if you can't remember exactly serum the over the කියලා, then you're going to get confused. Right? Serum albumin is a transport protein. Ovalbumin is a storage protein that is in eggs. Okay. If you want, you can write down. Storage, like ovalbumin is a storage protein, right? Then finally, eliminate foreign bodies immunoglobulins. Okay, no? Defensive protein. Oh. So in the second one also, there's a small section you can't do, no? With the nucleic acids, we can't do, no? It's very difficult to classify MCQs based on what you know, only what you know, right? So මේකත් අපි first දෙකෙන් අපිට answer එකක් ගන්න බැරි වේද? A protein with intermolecular bonds. So intermolecular comes after what stage? Inter molecule in the quaternary, right? In the quaternary you have intermolecular. Why? We get several molecules. So in the quaternary level, up to the tertiary level, you have only one molecule, right? Only one polypeptide chain, right? So the protein with intermolecular bonds, hemoglobin is good. Yes, hemoglobin is good. Collagen is also good. Why? Collagen is a quaternary structure. No, check the MC statement B. Monomer unit of a branched storage polysaccharide. Monomer. What are the branched storage polysaccharides? You know, branched monomer. Yes, glycogen is branched. Yes. What about starch? Starch is branched. දෙන්නකින් හැදලා තියෙන starch, right? Amylose and amylopectin. Go home and study. Okay. එතනින් මට මතක නැහැ කවුද who is branch amylose or amylopectin? Amylopectin is branch, no? Right? So starch qualifies, right? It's a branch storage polysaccharide, no? ඒතර දෙන්නෙක්ව ගන්න පුළුවන්. We can take glycogen and starch. Yeah. So monomer of a branch storage polysaccharide. අපි කවුරු ගත්තත් either glycogen or starch, who is the monomer? Glucose, right? Doesn't matter. An N base with one ring not present in deoxyribonucleotide is uracil. Okay, so the answer is uracil. So what is the best answer based on that? 3, no? But without even knowing the third one, you could get that, right? Because as the second answer, you have glucose only in the third statement. Get it? මම ඒක හිතලා නැහැ. No, that was like fortunately you can answer that, right? Okay, coming to structure quickly. Name an amphoteric molecule. Amphoteric. Both acidity and basicity, right? What is both acidity and basicity in the same molecule? Yes, Muta? Amino acid. Easy. Why is it called an amphoteric molecule? Write the definition. Both acidic and basic natures are found in the same molecule. Therefore, it's an amphoteric molecule. Three biologically important types of lipids. We know those. Yes. Triglycerol, phospholipids, and steroids. What is the composition of phospholipids? You know the list. What? Tell me. Recite. 2 fatty acids, 1 glycerol, 1 phosphate, 1 additional polar molecule such as choline, right? Choline nu thera වෙන වෙනම තියෙනවා. There are different oligophospholipid ඔන්නෝම තියෙනවා, right? But for your level, you only need to know choline. How does this molecule behave? How does it behave? For that, you can. I was hoping that you write how it forms the phospholipid. Sorry, how it forms the plasma membrane. Right? How does it form? Write down. When forming the plasma membrane, write down for the last structure they say, how does this molecule behave? What is this molecule? Phospholipids. When forming the plasma membrane, the hydrophilic heads face the aqueous environment. Aqueous means watery, right? Aqueous environment. Face the aqueous environment inside and outside the cell. To form phospholipid bilayer. Got it? මතකයි නේද bilayer එක? Right? Phospholipid bilayer. Okay. Any questions? No. Any questions? Next week, let's quickly finish nucleic acids. Then we can move on to the practical about microscopes. And I also have to finish about three practicals. We have one practical, identification of reducing sugars, then identification of starch. That's it. Okay. I'll give you a small separate tute for that. And yep. Microscopes. I'll try to bring a microscope. If not, what happened? Okay. In a school, you will definitely get the chance to handle microscopes, right? Definitely, there's going to be microscopes. And what else? Yeah. Any questions? No. Lakmal sir. Right. Right. You can go. Thank you for coming. And you can tell your friends. So I'm doing the next week session as free as well. Right? So ask your friends if they haven't got the practicals very well or understood, though, just come and see. Right? Okay. Thank you. Bye. Table 7. Anyone who needs the previous paper? Yes, I take on a card. I didn't give you. Okay. 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 Online students, thank you for joining.